Okay, so we'll hand over to Bipin for the first talk of the day, and in this session we'll have 3 talks before we have a break to have to go through the posters and have a little bit of a comfort break before a second session where we'll have three more speakers. So over to Bipin. Thank you.
Yeah, many thanks, Tim and Malcolm, providing this opportunity to discuss and present some of the recent work which we are carrying out at University of Nottingham. So the title of my presentation is Uncovering Novel Hormone-Based Mechanisms Controlling Root Responses to Soil Compaction Stress. So as we know that soil compaction is a modern agriculture problem, and this is because of the shift in tillage practices, which employs the modern heavy equipment. So as you can see in this map of the European agriculture land, nearly 2/3 of the entire arable land is prone to soil compaction. And while operating in the field, these big equipments like harvesters and combine machines, they press the topsoil as well as the subsoil, and this compaction basically inhibit the root growth elongation in compacted soil. So you can see in the left hand side the CT image of rice roots growing in non-compacted soil and branching pattern, whereas the rice root which is growing in compacted soil, it really struggled to penetrate in the hard soil and basically reduce the foraging ability of the plant, which ultimately inhibits or restrict the yield. So based on this modern agriculture problem, we recently discovered that how plant roots really sense the compaction stress. So what we found that basically a gaseous hormone called ethylene acts as a key signal to sense the soil compaction. So how does it sense the soil compaction? So as you can see here in the root soil interaction image using the non-invasive imaging, computed tomography imaging, both wild type and the ethylene insensitive mutant called IN2, they grow nicely in the non-compacted soil condition. Whereas in the compacted soil condition, the wild type roots really struggled to penetrate the hard soil, whereas the ethylene insensitive mutant behaves as if it is blind and can't sense the mechanical impedance imposed by the compacted soil condition. So based on this groundbreaking discovery, we proposed a model where the roots which is growing continuously in the non-compacted soil release the gaseous hormone ethylene, and this gaseous hormone ethylene diffuse away using the large air-filled pore networks. On the other hand, in the compacted soil condition, the soil pore volumes get very small and the network is broken down, so the gaseous hormone is restricted to diffuse away from the site of its production. So over the period of time, it accumulates near the root tip and basically enhance the ethylene response. And this heightened ethylene response in the root act as a stop signal and restrict the root tip elongation in the compacted soil. So based on this seminal knowledge, we were interested to know that how these root adaptive responses is controlled by ethylene. So particularly, the first root adaptive response which we were interested was the thickening response. So when you are growing the plants in compacted and non-compacted soil condition, you see a very contrasting difference. So when you grow,
Bipin, sorry to interrupt. Could whoever's not muted please mute their microphone? Thanks. Sorry, Bipin, carry on.
Yeah, it's fine. Thank you. So when you are growing the rice plant in the compacted soil condition, you see a very contrasting difference, that the cortical cell size becomes almost double or more than double in size, and it is due to the isodiametric expansion of the cortical cell size. So the diameter of the cortical cell is significantly increased, and we wanted to know and explore that which signal is controlling this response? Is it ethylene alone which is controlling this response? Or is there any other downstream signal which controls this thickening response of the root tips, which is growing in compacted soil condition? And to understand this, when we were profiling the expression of the most of the genes which are involved in the root responses, interestingly, we found that most of the ABA biosynthetic genes in rice were upregulated under compacted soil condition. So we wanted to confirm this, that whether actually the concentration of the ABA is also enhanced. And when we profiled the ABA level in the rice root tips growing in non-compacted soil condition, which is 1.1 bulk density, and in compacted soil condition, which is 1.6 bulk density, we found that indeed the ABA level is almost double in compacted soil condition. So, we were thinking that why the ABA level is increased in the compacted soil condition, and what is the advantage of having more ABA in the root tips, which is growing in compacted soil condition. And to explore that, we took advantage of a highly ABA deficient mutant called MHZ5, which is MAU-HUSI5, and basically it has around 80% less ABA level. And we found a very surprising result that this ABA deficient mutant was not showing the swelling response at all when growing in the compacted soil condition. So as you can see here that the swelling response of the ABA deficient mutant was abolished as compared to the wild type. So to understand the advantage of having less ABA or disadvantage, we did the CT imaging, whether it has any advantage in root penetration. And to understand this, when we did our computed tomography imaging, as you can see that the ABA deficient mutant MHZ5 has higher root penetration ability than the wild type. So this means that the plant roots, when not accumulating enough ABA in compacted soil condition, so basically they can penetrate much better than the wild type condition. So to understand that which root response is controlling this, we profile the other ABA mutants like ABA1, ABA2, and MHZ5, and we found that in almost all of the three different ABA biosynthetic mutants, the ABA level is not accumulating, and that's why we think that the roots remain narrow and they can penetrate the compacted soil better than the wild type. And to confirm this, we here imaged anatomically the radial expansion of the cortical cell in all of the three mutants. And as you can see here, that the ABA mutants, biosynthetic mutant, they are accumulating less ABA, and that's why they are not exhibiting the higher cortical cell expansion as compared to the wild type, and that's why it appears that may have higher root penetration ability than the compacted than the wild type. So to confirm this, we did the CT imaging of the other ABA biosynthetic mutant, including the MHZ5, and interestingly, all of the ABA biosynthetic mutant, including ABA1, ABA2, and MHZ4 and MHZ5, all of them exhibited higher root penetration ability than the wild type. So now we confirm that the ABA biosynthetic mutant, they do not accumulate enough ABA, and that's why the radial swelling response is reduced when growing in the compacted soil condition. And the advantage of this is that the root tips remains narrow and they can penetrate the compacted soil better than the wild type. So now we were more interested to know that how this the hormone wiring is controlling this root response, like whether the ethylene is upstream to the ABA signaling, or is the parallelly ABA is controlling this swelling response. So we did a very simple and elegant experiment where we exposed the wild type plant roots with ethylene. As you can see here on the right hand side, you see the radial expansion of the root cortical cells after ethylene treatment. And similarly, when we treated the plant roots with the ABA, we again saw the similar radial expansion of the cortical cells. So we were puzzled to see this, that which one is actually controlling this radial swelling response. Is it the ABA or is it the ethylene which is controlling this? And to nail this out, we took advantage of the ABA biosynthetic mutant. And interestingly, when we did the ethylene treatment to the ABA biosynthetic mutant, we didn't see the swelling response at all. So this confirms that ABA is really critical for radial expansion of the cortical cell, and it acts as a downstream signal to ethylene. To go beyond a little bit, we also employed the ethylene insensitive mutant IL-1 and IN2. And as you can see here, because they are ethylene insensitive mutant, they didn't show swelling response, whereas the wild type plant roots was showing massive increase in their cortical cell diameter. And interestingly, when we use this mutant for ABA treatment, you can see here that both the ethylene insensitive mutant IL-1 and IL-2, they exhibited radial cell expansion response. And this experiment confirmed that indeed ABA is critically required for radial expansion of the cortical cell, and ABA must act as a downstream signal to ethylene signaling pathway in compaction. So based on this action of the ABA acting as a downstream signal and controlling the radial expansion of the cortical cell, we were interested to know the second classical root responses which we see in the compaction. And the second response is that as you increase the compaction response or the swell bulk density, you see the higher reduction in the root growth. So which signal is controlling this root growth in compaction? And to underpin this, we employed our Raisa auxin reporter line. And we got a very interesting result that when this auxin reporter is growing into the compaction condition, you see two important information. First, there is a massive increase in the radial swelling, as if you can see, compare the two images, and there is a higher fluorescence intensity of the venous in the root tip. And as you go a bit further up in the elongation zone, you can massively see the higher fluorescence signal, indicating that auxin level is increased in the epidermal cells. So what will happen when the auxin level is increased in the epidermal cells in compacted swell condition? Basically, enhanced auxin response or label in the epidermal cell will reduce its axial expansion, and which will inhibit the elongation of the root tip in compacted swell condition. So whether it is actually controlled or regulated by the ethylene, we crossed the DR5 venous Raisa reporter line to the IN2 mutant. And as you can clearly see that we were only observe the vascular signal. The epidermal signal is completely abolished irrespective of the compaction response. So what does this mean is that ethylene is directly regulating the higher auxin response in the compaction, and this higher auxin response in the epidermal cell is controlling the epidermal cell expansion in compacted swell condition. And so we took advantage of another auxin transport mutant called AX1, which is basically ethylene insensitive as well, and which controls the auxin transport from the root tip to the elongation zone. And when we grew the AX1 mutant, both the alleles in non-compacted and compacted swell condition, we observed that the auxin transport mutant, AX1, has a relatively higher root penetration ability than the wild type. So it confirmed that the mutant is unable to transport back the auxin from root tip to the elongation zone, and that's why the epidermal cells have a reduced auxin level, and this is the region why the epidermal cells remains able to elongate and expand axially, and that's why the AXON mutant is able to penetrate higher than the wild type. And as you can see here clearly, when we cross the auxin reporter line with the AXON mutant, then the backflow of the auxin is completely abolished, irrespective of the compaction response, whether it is non-compacted or compacted level. The auxin is unable to move back to the elongation zone, and that's why there is no impact of higher auxin accumulation in the epidermal cells to reduce the cell expansion. And finally, they can be thought that could it be that the auxin level is also increased during compaction level. And to extrapolate this, we use the auxin biosynthetic mutant UC8 and employed the CT imaging. And interestingly, we found that the auxin biosynthetic mutant again showed higher root penetration ability than the wild type. And this is because now there is no enough auxin synthesized at the root tip, so its transport back to the elongation zone is also reduced, and that's why the epidermal cells remains able to elongate and expand, and which helps the root tip to penetrate and elongate into the hard soil. So based on these two responses, distinctly controlled by the ABA and auxin, we came to a model where, under non-compacted soil condition, the gaseous hormone ethylene is diffused away and flushed out using the large air-filled soil pore networks, whereas in the compacted soil condition, the diffusion of the gaseous hormone ethylene is blocked, and over the period of time it accumulates near the root tip and enhance the ethylene response in the root, which directly or indirectly controls the ABA biosynthesis. And then this higher ABA level causes the increased radial expansion of the root cortical cell, which basically inhibit the root tip elongation in compacted soil condition. And parallelly, the higher ethylene response directly controls the auxin biosynthesis and enhance the auxin level, and then it moved or transported back to the elongation zone via AX1, which basically enhance the auxin level in the epidermal cell, which reduces the epidermal cell expansion. So integrating these two classical root responses, we came to this model that ethylene as a gaseous hormone is orchestrating two very important hormone signals, ABA and auxin, to control two distinct root responses synergistically. And finally, I would like to thank Professor Malcolm Bennett from the University of Nottingham, whose important role cannot be, his role was exceptional in shaping these root responses research work, and along with Dabing Chang, who critically provided several of the mutant and the genetic resources, along with Guqiang and Ajad Poonam and Michael Karaji, who did the hormone profiling from Czech, and Craig Struck, whose expertise in the CT imaging, and the great mind from Professor Sasamuni at the root soil interaction, and Professor Jan Tutt and Professor Robert Saab. So entire team critically involved and shaped this root research very well. And in the last, I would also like to thank and acknowledge the funding agencies, particularly BBSRC and the Raisa Society and the Future Food Beacon, who helped us a lot to carry out these important research work. And thank you everyone for listening. And now I'm ready to answer your questions.
Thanks, Bipin. That was a fascinating talk and a great way to start the afternoon. So we have a couple of questions in the chat, and they're on the same sort of lines about the interaction with water. So Paul, I'll ask both questions and maybe you can answer both of them in one. So we have a question from Paul Hallett, which is, have you explored the combined impacts of mechanical and water stress? Could be interesting. There could be interesting diffusion impacts. And Glyn Bengoff asks, says, interesting talk. Were your experiments performed at field capacity? If so, what happens in hard, dry soil where ethylene diffusion is not limiting? So I think they're both of a similar sort of question. So I wondered if you could comment on the interaction with water at all.
Yeah, both of the questions are really important. So to the first one, we did all the experiment at the field capacity level, and we know that the solubility of the ethylene in water is much less, around 10,000 times lesser than the air. So we suspect that if we increase the water level in the soil pores, of course the diffusion would be much higher, but we also know that water acts as a lubricant. So the strength of the soil can be reduced. But we have never done such an experiment where we combined both the water and the mechanical impedance. I think it would be really fun to do that. The second question was related to the hardness of the soil. So we know that that the ABA level is increased in drought, and we also found that the ABA level is increased in compaction. So we think that in compacted soil condition, the soil pore volume is highly reduced, and basically the roots has to exert much pressure to extract the water from the tiny pores, and that's why the plant roots sense like moderate drought conditions. So even though there is a water, it's a much harder for the root to extract the water from the tiny soil pores. So when you make the soil a little bit dry, then diffusion can be much easier for them because there is less water. But of course, the strength of the soil is again increased. So we think that the combined action of both would have a different consequences. Yeah.
Okay, thanks, Bipin. There's a few questions coming in now, so we probably won't have time to look at them all. So maybe afterwards you can have a look in the chat and answer some of the questions you don't get a chance to. There's a question from Leah Band. I wondered if the cortical radial expansion is a parallel and independent response to the reduced longitudinal growth. What is the advantage of radial expansion? Why do roots do this?
It's a very interesting question, and we thought a lot that why the plant's roots are showing this swelling response. And last week, I was in a meeting in rice functional genomics where one of the South Korean professors showed that if you put the ABA to the plant roots, basically you increase the cortical cell size, and you also store the water and the nutrients. So I suspect that when the plant roots become thicker, it conserves the water and the nutrient to cope the hard soil's condition.
Okay, I mean, I had a similar question in a way that actually bigger cells are cheaper to produce. So a root system might want to produce bigger cells, and therefore, what's the trade-off between cheaper roots and penetration ability?
Yeah, that's also a very interesting question. So we found that if the root or the mutant, if when they are not showing the swelling response, so basically they are narrower, and that helps the root tip to penetrate in the hard soil. So yeah, so putting when the genotypes are the mutant, when they are not expanding radially, that means they can penetrate the hard soil in a much effective way than the one which are showing higher swelling response. So I think cheaper strategy would work very well in this case as well.
Great, thanks, Bipin. In the interest of time, we'll probably move on, but there are some more questions. So there's a question from Mike Lucas, Adam Price, and Johannes Postma in the chat. If you would like to follow those up during the next few minutes, that'd be great.
Yeah, so I can answer
if you stop
questions.
Yeah, if you answer those in the chat, that'd be great. If you can stop sharing now, and we'll move on to the next speaker. So thanks ever so much, Bipin. That was really great.
Yeah, thank you.
So the next speaker is Dr. Rahul Bhosale, who's also from the University of Nottingham, and he's going to be presenting some of his recent research on discovering how cereal roots control their growth angle using a novel anti-gravitropic offset mechanism. So Rahul's already sharing his screen. Maybe you can show us that one. That's the one. So that's Perfect, and ready to go whenever you are, Rahul. Thank you.
Okay, can you hear me?
We can hear you perfectly, yes.
Okay, great. It's a great pleasure to be here. And again, thanks Malcolm and Tim for the opportunity to talk about our latest work, where we discovered a gene called EGT1, or we named it enhanced gravitropism 1, which controls root growth angle in cereal crops by employing a novel anti-gravitropic mechanism. Okay, I think slides moved to the next one. Tim?
Yeah, yes, that's
Okay.
perfect.
Great, thanks. So root angle determines the region of soil where the roots will forage and capture soil resources. For instance, if you have a steeper root system, so they are capable of capturing mobile resources such as water and nutrient from deep soil. And they are also considered important in burying carbon in deeper soil in sustainable manner. On the other side, if you have a shallow root system, which they are capable of capturing immobile resources such as phosphate from the topsoil region, and also they are considered to be well adapted to waterlogging or hypoxic conditions. So thus, the root angle greatly influences the region where the soil resources will be captured and confers tolerance to some abiotic stresses. So thus, understanding genes and mechanisms controlling root angle is important for crop improvement, not only to develop resilient varieties to abiotic stresses and improve productivity, but also to devise some strategies to bury carbon deeper in soil in sustainable manner to mitigate global carbon burden. So if you look at the cereal crop root system, which includes different root classes such as primary, seminal, crown, and lateral roots. So they usually are, these root classes emerge and are maintained at different set point angles. So they are called gravitropic set point angles. So for instance, you have the primary root, which grows towards along with the gravity vectors downside. And you have different root classes like seminal and crown lateral, so they are maintained at different set point angles. So they are usually considered to be regulated by two competing mechanisms, so called gravitropic response machinery, and the other one is the anti-gravitropic offset machinery. So one nice way to visualize these two competing mechanisms is just vertically growing plants. You can turn them at a particular set point angle by turning them 90 degrees. And then you can see the primary roots bends much faster because there is no anti-gravitropic component involved. But the seminal roots bend much slower because of the competing gravitropic versus anti-gravitropic mechanisms. And after some point of bending response, so these primary and seminal roots go back to their original set point angle, suggesting that so when they emerge, so there is a set point angle is determined, and after bending response as well, this is they go back to the original set point angle, meaning that they are maintained during their growth as well. So a lot has been known in terms of the gravitropics or regulatory components involved in gravitropics machinery. Is mainly a lot of work done in the past in our offices, but very little is known about the anti-gravitropic machinery. This could be one of the reasons could be the main root classes that do not exist in our process, such as the seminal and crown roots. And so there is a knowledge gap about regulatory components involved in this anti-gravitropic offset mechanisms, especially in cereal crops. So to bridge this knowledge gap, what we did, we screened a tilling mutant population in Morix cultivar using a semi-hydroponic growth pouch system. And we identified two interesting mutants which show a very steep root angle. So for instance, one of them is the TM194. So you can see that compared to the Morix wild type, the seminal roots are much steeper. So we confirmed this phenotype directly in soil using X-ray micro-CT, and then the TM194 mutant consistently shows a very steep seminal angle. So when we did X-ray micro-CT at different time points, such as the 20-day seedling and grain maturation stage, so we could see that the other root classes, such as lateral roots as well as the crown roots, are much steeper in the mutant. So overall, this suggests that this mutant, all of the classes are going steeper, and most likely because of the no anti-gravitropic component here. So to pinpoint the locus, which is responsible for or the mutated locus, which is responsible for the steeple root angle. So what we did, we crossed the TM194 mutant in a parquet cultivar, so which shows a very similar root angle as the wild type. So F1 progenies, so they all behave like a wild type, but in F2 we saw a clear segregation of 3s to 1. So one recessive allele basically showing you a steeper root angle phenotype. So we used the same F2 population to do a bulk segregant analysis and map a mutated locus, and it was on the chromosome 6. So there is a 130 MB region, which is, you can see it very nicely here. And so there are only 4 genes within this span of region which has G2A mutation, which is expected because of the mutagenesis. And but we still don't know which is the gene is responsible for this for the root angle phenotype. To address this, so we looked at the other mutant. It's again from the Morex Tilling mutant population, which shows very similar phenotype as the TM194. And it also showed seminal root as well as lateral roots phenotype, which is consistent with the TM194. So it suggested that, or hinted that, probably this mutation is also within the same gene. So what we did to confirm this, so we crossed TM194 and TM3580 mutant alleles, and we saw that the F1 progenies do not complement phenotype. So this clearly showed that. So both TM194 and TM358 mutants highlight the same mutant locus. So we exome capture sequencing we performed this on the TM3580 mutant, and we saw within this 130 MB region of the bulk segregant analysis, only one overlapping gene is present, which shows G2A mutation. And so this gene encodes a F-box and tubidomain-like protein, and that the mutant TM194 mutant is within the tubi1-like domain, while the TM3580 is predicted to as a splice acceptor variant. So as expected, these two mutants are point mutations. So we performed transcriptome analysis as well as qPCR analysis, and as expected, we saw that there is no change in expression or significant change in expression of this gene in both the mutant alleles. So this suggested that the phenotype is likely due to altered protein structure. So to gain some understanding about how these mutations affect the protein structure function, so we performed homology modeling for this TM194 mutant, and so we observed that the 395 residue where the G2A mutation is, it sits within a negatively charged, positively charged domain, which is stabilized by the negatively charged C terminus. But this G2E substitution is from small neutral to large negatively charged amino acid substitution, which is basically going to disrupt the positively charged pocket. So this likely would have an impact on the protein structure and function. So we also looked at the this region where this mutation is, and it was interesting to see that there is a so this mutation is a 395 and 391 to 397. So there is a very highly conserved motif across 37 plant species we study. So all of these FW domain-like genes or encoding genes, this is very highly conserved motif, and the mutation is within that. So this suggested that maybe this is likely the reason why we see such a striking phenotype. Similarly, as we know that the TM3580 is predicted as a splice acceptor mutation, so we performed transcriptome analysis. We mapped the transcripts to the reference genome as well as the Morex, and we saw that indeed the TM3580 is a splice acceptor variant, and which has a deletion of 9 amino acids. So this mutation is not within the tubby domain, it's closer to a box domain. So we wondered whether does it have any implication in protein structure function. So to do this, we used AlphaFold to predict the box and tubby domain or the whole protein structure. And so we saw that the box domain has a partly charged and partly hydrophobic protein-protein interaction surface here. And the wild type tubby domain very nicely complements this protein-protein interaction surface domain. But the mutated one, which kind of introduces alpha helix alpha helix, which causes a juxtaposition of negatively charged residues in this protein interaction surface, which is likely to disrupt the whole protein-protein interactions between, or the domain interactions between FBOX and TM358 FBOX and the tubby domain. So this would have an impact on the protein structure and function. So this suggested that, so we got two mutant alleles, which shows a very steeper root angle phenotype in all root classes, and they highlight the same gene, which encodes for a FBOX and tubby domain protein. So both mutant alleles don't show any change in gene expression, but show altered protein structure function. And so overall, we concluded that this is the gene which is responsible for steeper root angle phenotype. And because of its enhanced gravitropic phenotype, we called it as enhanced gravitropism 1. So next, we wondered, because this gene seems to be an important in terms of controlling root angle in all root classes. And if you mutate it, you get such a striking phenotype. So we wondered whether the allelic variation between this gene could explain the natural variation in root angle in barley germplasm. So we took advantage of a Wilby collection which is already have exome sequencing data available. So this is a Wilby collection, is a part of wheat and barley legacy for breeding improvement collection. So we took more than 450 barley barley accessions, which is basically different cultivars, land races, wild and breeding lines. And so we found that there were there were 6 haplotypes within this gene. So 2 haplotypes basically resulted into a missense substitution. One of the haplotype 2 had a narrow root angle, while the haplotype 4 had a wide root angle phenotypes. You can see it here. So interesting thing is the haplotype 2 is actually just 4 amino acids upstream of the mutation that we identified in TM94, and this haplotype is actually, the mutation is within the conserved motif. So this suggested that, so actually EGT1 could be a hotspot for natural variation in root angle in barley population. So we wondered whether this would also be, is this a very specific to barley, or is the function of EGT1 is also likely to be conserved in other cereal crops? So when we looked at different orthologous genes from selected cereals like in rice, maize, brachypodium, and wheat, so we saw that there were, so this EGT1 like genes form a very distinct clade, and so we looked at, so to prove that whether this is a gene is function is conserved in other cereals. So we identified T-link mutant lines in tetraploid wheat on A and D genome. So single mutants did not show any, homozygous single mutants did not show any phenotype. But when we made a double mutant phenotype, so did double mutant, which showed a striking, again similar to barley, a very steep root angle phenotype in seminal root as well as the lateral roots. So this suggested that the EGT1 function seems to be conserved in wheat, and it's because of it forms a distinct clade in with other cereal genes. So we thought it could be conserved in other cereals as well. So to confirm whether this gene function in gravitropic or anti-gravitropic offset mechanism. So what we did, so we hypothesized that if the gene is involved in gravitropism, so you would have a gravitropic defect. But if your gene is involved in anti-gravitropic offset mechanism, if you disrupt that, so because of only gravitropic component, your roots will bend much faster during the gravitropic bending response assay. So what we did is like we grew Morex and TM194 mutants vertically, and then turn them at 30 degrees, 60 degree and 90 degree set point angles. And what we saw that as this set point angle changed from 30 to 90, so the mutant the bending velocity of the mutant was much higher than the wild type, and it increased from 30 to 60 to 90 degree. So you can see in the representative images, even in 0.5 or half an hour, you can see Morex, it doesn't even start, or it start to bend, while the TM194, the root tip, is already started or had some bending response. So this suggested that EGT could be involved in anti-gravitropic offset mechanism to control root angle in cereal crops. As we know that the auxin plays an important role in influencing both gravitropic as well as anti-gravitropic offset mechanism. So we wanted to understand whether the gravitropic machinery is intact, and whether exogenous application of NAA auxin, or by inhibiting the auxin transport, we would see any impact on the bending response. So it seems like the wild type and the mutant behave exactly the same, suggesting that the auxin-dependent gravitropic machinery is intact in EGT1. It seems, again, confirming that it's mainly the impact is only on the anti-gravitropic offset machinery, not on the gravitropic machinery. So we wondered whether, because auxin has been already known to be involved in both mechanisms, so we wondered whether the EGT1 is influenced by auxin levels, or it is downstream of auxin. So we checked the promoter of the EGT1, and we did not find any auxin response elements, which is important for inducing the expression in auxin-dependent manner. Using QRT-PCR, we also confirmed that there is no significant increase in EGT1 expression even after 8 hours or 4 hours or 8 hours of treatments, while the I36, which is auxin-inducible gene, which has a significant increase in expression. So this suggested that, so we have found a gene which is responsible for controlling root growth angle, which functions in a anti-gravitropic offset mechanism and is not influenced by auxin, and it is independent of auxin. So next, we wanted to gain a little bit more understanding about how this gene controls the root growth angles. So to understand the downstream processes which are regulated by EGT1. So we performed a transcriptome analysis on this on a 4-day-old seedlings and 1 centimeter, sorry, this should be 1 centimeter root tip from all seminal roots. And so we extracted RNA, we performed RNA sequencing. So we looked at differentially expressed genes between Morex and the TM194, as well as the Morex versus TM3580. So we only looked at the genes that are commonly differentially regulated between both mutant versus the wild type, and we performed a co-enrichment analysis. So what I'm presenting here is just a pruned version of it, but we did not see any enrichment for auxin signaling components as expected. But strikingly, there were only 2 categories were mainly perturbed in the mutants versus the wild type. And those 2 categories involved in response to oxidative stress and cell wall organogenesis or biogenesis. So to confirm that whether indeed these processes are affected in the mutant, so we first looked at the ROS levels. So we used a fluorescent probe to detect the ROS levels in the root tips in Morex and the TM194 mutant. And we saw that indeed the ROS levels are reduced in the TM194 mutant, mainly in the meristem and the elongation zone. So as ROS has been linked with the cross-linking cell wall material and increasing the stiffness in the cell, so we wondered whether the reduction in ROS levels would have an impact on the stiffness, cell wall stiffness. And as we see in our transcriptome data set as well, so the other category which was really perturbed was the cell wall biogenesis and organogenesis. So we performed atomic force microscopy experiments in meristem and elongation zone into 3x3 kind of array. And so we calculated apparent stiffness, which is basically force observable compared to the distance curve. And so this is what you are looking at here is a cross section, not cross section, longitudinal section of the root, which was probed for atomic by atomic force microscopy. So what we saw overall, there is a great reduction in cell wall stiffness in the TM194, but when we divided this capture data into inner steel vascular tissues versus the cortex tissue, so we saw that there is no stiffness difference in the steel, but there is a significant stiffness difference between cortex in the wild type versus the mutant.
Actually,
So
Bipin
suggested
sir
that. So TM194 mutant overall shows the reduced stiffness in the root tip, but mainly in the root cortex. So we wondered whether this gene is specifically expressed in the cortex or in other tissue types. So we performed RNA in situ hybridization using anti-seps probes. So we saw that the EGT1 is mainly expressed in the elongating cells and mainly in the steel region. So there is also some expression detected in the cortex and epidermal cells. So what we see here is that we got a gene which is mainly expressed in inner tissue, but while the stiffness differs in the outer tissue. So it's possible that the EGT could function directly in the cortex to control the reactive oxygen species processes or the cell wall stiffness processes, or it moves from inner to the outer tissue. So based on this, so what we put forward in a hypothesis now that so EGT1 could actually regulate the peroxidases and cell wall loosening related enzymes to control the cell wall stiffness in cortex, to control the antigravitropic response machinery and root growth angle. So with this, I would like to thank many people involved in this project, especially thanks to Rick, Serena, and Mia, who perform most of the work, and also Malcolm and Silvio for co-leading this project. And also to funders, BBSSA Discovery Fellowship, ARPA-E, on which Ricardo was mainly funded on, European Plant Phenotyping Network funding, International Wheat Partnership funding, which funded the screening of this mutant lines, and also University of Nottingham Future Food Beacon for supporting me. Thank you so much. I'm very happy to answer any questions.
Thanks, Rahul. That's really great. Again, a really nice, elegant demonstration of some fantastic fundamental root biology. So, yeah, we've got time for a couple of questions. We have one in the chat from Doris Vetterlein, which is, any indications that the mechanism you observe for root tips of these very young seedlings have the same importance for root tips within root systems of older plants, past tillering, heading, flowering, etc.?
So we looked at So of course we did not look at the root systems later on in the later developmental stages, but it's very clear from the X-ray microCT imaging data that all the roots are just going downwards. So they don't have this anti-gravitropic offset mechanism, so they are really gravitropics. And so even in the later developmental stages, we would see that the roots are going downwards. Yeah. And so this again, so I'm just showing you data here, whether we looked at the shoot and leaf angle phenotypes, and so this mutation seems to be, or the gene seems to be really root specific.
Okay, and I think that answers the next question as well. But I have another question, which is, you showed some barley haplotypes which had a relatively small difference in angle, so it's 10 degrees or so between the haplotypes. So I just wondered, are you able to see a difference in the functionality of those haplotypes? Are they better? Are they more drought tolerant? Are they able to get phosphorus more easily or whatever? So does that small change in angle make a difference?
Yeah, so this is a great question. So of course, this is the kind of next work we want to perform. So to understand, so what's the relevance of this having a steeper root angle? So of course, in literature, we know that steeper root angle, they confer tolerance to drought and the nitrogen stress. But we haven't looked at the haplotypes in barley population, whether they are drought tolerant or not.
Sounds interesting. So just finally a comment from Adam Price, which is that in rice they have QTLs on top of this gene for root growth in general and for gravitropism specifically. So it might be interesting to follow that up in rice as well.
Yeah. So we created a CRISPR edits in rice. So we're just waiting for homozygous lines to see the phenotype. Yeah.
Excellent. Thanks, Rahul. So we'll move on to the next speaker now. So if you can stop sharing, we'll move on to Dhin Islam, who is from the University of Aberdeen. And he's going to be presenting some of his latest research on the influence of biopores on the performance of rice. So
Tim, I think we have the same problem as yesterday, so I can't.
Okay, so so Dhin, if you start sharing, it should bump Rahul off if you can.
Great, thank you. Here is the stop sharing here.
Yeah.
I think.
Yeah, no, we can see that. We can see it perfectly, and we can hear you perfectly. So whenever you're ready, you can start.
Okay. Thank you, Tim, for your introduction. And today I'm going to talk about a part of my research that I did during my ongoing PhD project. Rice is one of the major food crop in the world, but this crop faces many challenges. So what are the major challenges in rice production? First of all, fresh water shortage, because most of the rice is produced under flooded condition and it requires 2 to 4 times higher water than any other upland crops. Most of the farmers in rice growing countries uses intensive tillage practices to make the land fertile during transplantation. It increases the cost of production and destroy the soil structure, as well as it creates subsoil compaction problems. In the rice field, there is another major problem, greenhouse gas emission, which increase the global warming. Recently, labor shortage is going to be alarming for the rice cultivation around the world. So scientists are trying to develop different types of water saving technologies, conversion of flooded rice into AWD, offline to offline, as well as aerobic. This tackle the problem of water shortage as well as greenhouse gas emission from the rice field. Most of the rice growing countries farmers use different techniques with multiple rice crop over the lands with growing condition alternating to flooding to AWD or even aerobic. But the main problem under upland or aerobic rice cultivation after flooded is the subsoil compactions. So what are the consequences of subsoil compactions? It reduces the macroporosity as well as hydraulic conductivity, aeration, so that root can't grow into the deeper soil. So most of the subsoil resources remain unutilized by the rice root if the subsoil is compacted. So that subsoil compaction is a big issue. So how can we mitigate subsoil compaction? There are two main ways by which we can mitigate the subsoil compaction. Deep tillage is one of them, and it's most widely used technique, but it's costly, as well as the effect of deep tillage is short-lived, so that we can use Bipur for
The sound is
mitigating
still
the
not
subsoil
there.
compaction effect in a sustainable way. So what's our Bipur? Bipur are the void in the
But
soil that
obviously,
are
nobody else
created
has
by
the
the
problem because
soil lives, mainly by the plant roots and the earthworms. So why are Bipur so important for mitigating the subsoil compaction problem in a sustainable way? Bipur can provide preferential pathways for the root to go into the deeper soils, and it increases the aeration, microbial activities, as well as nutrient releasing capability of the soil. Finally, the effects of Bipur are long-lived. So what's our main focus is where having influence of macropore on the root growth through plough-in. What would be the differences of growth of root between the deep and shallow rooting rice cultivar and grow in macropore seated core? How would we have one grow root growth through different types of macropores? Have any impacts of naturally created macropore on the growth of rice root? So for answering these questions, we did two experiments. In our first experiment, we used 10.2 centimeter diameter and 48 centimeter height core. Top 5 centimeter was empty, and then 20 centimeter topsoil was packed loosely, and 5 centimeter was plough-in, and then rest 18 centimeter was the loose soil, just like topsoil. We artificially inserted different macropore treatment through the plough-in. Our treatments were the control plough-in as well as Bipur cracks in combination of Bipur and cracks. We used two contrasting root architectural genotypes, IR64 shell rooting and black gora deep rooting, and we measured both above ground and below ground parameters. And we also scanned through X-ray CT during harvesting. For more detail, you can go through our papers. And although the penetration, resistance, and bulk density across our treatment were same, we found that macropore-treated cores had great impact on the root growth of rice than the control treatment. Here you can see the rootland density and the number of roots are higher for the macropore treated cores than that of the control treatments. The better performance under the macropore treated cores might be due to plant root can sense their environment and prefer to grow through the most favorable root zone. And as you know that the biopore or macropore can improve the soil aeration, microbial activity, as well as nutrient releasing capability of the soil. So what's about macropore shape? As you know that in our experiment, we used two types of macropore, biopore and cracks. Although the volume of Bipur and crack in our treatment was same, we found that macro crack were better for the root growth through the plough-in than the Bipur. Here you can see rootland density in the plough-in was better for the crack treatment than that of Bipur treatment. In case of root number, also the same effect we found. So the better performance under the crack treatment through the plough-in because of cracks are thinner and the surface area was higher than Bipur, so that root soil contact was higher in case of the crack treatment than that of bipod treatment. But if you go into the subsoil, you can see the crack treatment perform lower than that of bipod treatment. But it's because of we saw that at the bottom surface of the plow pen, there we saw the bipod was more crowded with the root. So when this crowded root reaches to the low subsoil, it can proliferate profoundly. As you know that we used to different root architectural genotype, and the deep rooting black gora benefited more by the presence of either bipod or cracks. Here you can see the root length density as well as the number of roots are higher in case of the in case of deep rooting rice genotype than that of shallow rooting rice genotype IR64. So it might be due to the deep rooting black gora has more stiffer root angle as well as stronger gravitavism, and it has greater root branching capacity. So there was a higher chance to finding out the macropore by the black gora than that of IR64 rice genotypes. As you know that in our first experiment, we used artificial macropore, but you know, artificial macropore as well as natural macropores are different. So what will happen in natural biopores? For answering this, we did another experiment with natural biopores with the same core, but in this time we used a little bit different treatment. Here you can see there was a control treatment, soft plough-fan and hard plough-fan. In this time we used it was a double cup experiment, and in the first season we used IR64 and black gora in flooded conditions, and there was a control with the wood plant, and the growing duration was 4 weeks. After during the harvesting we cut the shoot at the soil surface level and then kept it into the greenhouse for decomposition, and our decomposition period was 1 month, and then we cultivated Breedon 28 on the same core in the second season in upland conditions. And we found that in the flooded season, the number of roots at the bottom surface of the plough-fan was higher in case of deep rooting black gora than that of IR64, and then when it's decomposed during the decomposition period, it creates more biopore than the IR64 one. And we also and it is very interesting that in the upland season we found that the overall root land density was not affected by the presence of plough-fan. It's because of the biofilm created by the first season rice root that improves sardine cell mechanism by providing less mechanical barriers, better variation, and improved water and nutrient uptake from the deeper soil. In conclusion, we can say that the presence of macropore greatly improved rice root growth through the plough-fan, deep rooting variety being far more responsive to macropore presence. Not only did the presence of macropore influence deep rooting, but also macropore shape deep rooting genotype helps more biopore formation than shallow rooting rice variety. Previous season rice root reduced the effect of plough in the next season rice cultivation by providing root growth channel. In our next work, at this moment, we are doing XCT image analysis that we scan during the artificial and natural biopore experiment, and we have already set up an experiment for exploring the rhizosphere development under AWD in rice cultivation. Maybe hopefully we will get some good result. This is my acknowledgement slide. I'm highly grateful to Professor Paul de Hallet and Professor Adam Price, and also all of the members of Aberdeenshire Biophysics Group. I'm highly grateful to University of Aberdeen, my university in Bangladesh, and my funding organization, Commonwealth Scholarship Commission in the UK. That's all. Thank you.
Thanks,
I'm very
Dhin.
happy to take any questions.
Thanks, Dhin. That's really great. You've left us plenty of time for questions, and there's a couple of questions in the chat, which I'll ask you now. And if anyone else has got any more, well, there's a few coming through now. So the first question is from Dr. Satpute, I think. RLD, or root length density itself, is not an important parameter of RSA. Can it be replaced by root thickness?
Can you repeat the questions, please?
So root length density itself is not an important parameter of root surface area, or RSA. Can RLD be replaced by root thickness?
Yes, we also measured the root diameter, and we also found the same result. It was higher in case of deep rooting Blagora than that of IR64. Yeah, we have better result as well.
Great. A question from Malcolm Bennett. Do the deeper rooting varieties show differences in hormone sensitivity to signals like ethylene?
I think for the deep rooting Blagora, you know that deep rooting Blagora has a stiffer root angle as well as strong capitophysium and higher branching capacity. So deep rooting rice variety can easily find the biopores or macropore so that they can easily go into the deeper soil, and then they can utilize the subsoil resources. Yeah.
Okay. There's a question from Mike Lucas. Do you think that the additional roots growing through the pan with biopores all take the easy route through the biopore, or is there increased growth through the soil matrix as well?
I think can you repeat the questions, please?
So basically, do all the roots go through the biopores, or do some roots also grow through the soil matrix in the plough pan?
Okay. So in case of the root grow through the biopore, we identified by using X-ray CT. We scanned the X-ray. We scanned the bottom surface of the plough fan by X-ray CT, and we found that in both season, for the first season as well as for the second season, we found that most of the root use the same root position, so we can confirm that is root growth or the growth channel by the previously created by the roots.
Great, thank you. I have a question also, which is So there's obviously a benefit to root growth of the biopores, and I just wonder how much of that benefit is down to relief of soil physical conditions, but how much of it is down to changes in chemistry, for example, oxygenation of the soil around the biopore, for example, might change things.
Yes. So I did not measure the oxygen concentration in our experiment, but I found some paper that did the oxygen concentration measurement, and they found that the oxygen concentration was higher in case of the biopore treated one, and it significantly impacted the root growth as well as the yield. And for the soil, for the nutrient concentration, I also found some paper in that they mentioned that the nutrient concentration was also higher for the biopore-treated course, because you know that mineralization is different on the aeration, proper aeration. So in case of the biopore-treated course, aeration is good, so that mineralization will be high. So yeah.
Great, thank you. One final question from Jambo Shen. I think biopores may be more important for upland crops compared with flooded rice. Why use rice?
Yes, it's really important for the upland season, because for the flooded one, flooded season, usually plant roots don't need to go move deeper, because the water is available on the surface. So, and there is a problem in the rice field. You know, most of the farmer use puddling during transplantation. So due to puddling, it creates soft soil compaction. That means plow fan that's below the plow layer. So when we grow rice in upland condition after flooded, then the plant need more water from the deeper soil, so that a plant root need to go into the deeper soil. That's why biopore is more important in the upland crop than that of flooded conditions. Yeah.
Great. Thanks ever so much, Dhin. Great talk and great answers to questions. So let's thank Dhin and all the other speakers for this first session with a virtual hand clap. The first speaker is Raisa Osama, who is from the James Hutton Institute, and she's going to be presenting some of her work on the interaction between raspberry roots and Phytophthora rubi. So Raisa, if you're ready to start sharing, we'll make a start with the second session.
Okay, can you hear me?
I can hear you fine, yeah.
Okay, I'll start sharing my screen.
And as before in the session, I will put my video on with a minute or so to go, so you know that you're nearing the end of your time slot. So that's...
So are you able to see my screen?
That looks perfect, yeah, absolutely. So Raisa, whenever you're ready, over to you.
Thank you very much. So I am Raisa Osama. I am a PhD student of the University of Warwick, but I'm based at the James Hutton Institute in Dundee. So for my PhD, I have been working with raspberry root rot, trying to understand the mechanism of resistance in the roots of the host plant. So Phytophthora root rot is one of the most important diseases of red raspberries, causing devastating loss to the raspberry industry. Most commercially grown raspberries are infected with this disease. It can cause up to 100% mortality of the host plant. As you can see from the distribution map, that this problem is not only a problem in the UK, but it is a worldwide problem. So the life cycle of Phytophthora may start from an oospore, which remains as dormant structures in the soil and that can survive in the soil over a long period of time. So these oospores germinate to produce mycelia and sporangia. So the head of the sporangia is filled with spores called zoospores, which when released can swim to the roots of the host plant, attach to it, and penetrate directly through by forming this infection peg. So this is one of the advantage of oomycetes, is that they don't need any opening or wound to enter the host plant. They can penetrate directly through the cuticles of the host plants. So after penetration, they develop inside the root intercellularly, and when they reach the vascular bundle, they uptake nutrients from these roots and then kill the roots. And then they continue their life cycle by producing mycelia, sporangia, and oospores again. So the most common method to protect raspberry plants from this disease is by growing them in plastic pots. More than 70% of raspberries are grown in this method, which is not economically or environmentally feasible. There are some fungicides that are available to treat Phytophthora root rot. However, they are being withdrawn to be used on raspberries. So there is no fungicide that is specifically available for raspberry root rot. So a long-term solution to this problem is to be able to develop resistant varieties that can be grown in contaminated fields without any economic loss. And in order to achieve this, it is important to understand the mechanism of resistance in the host plant. Here, this is two cultivars of the raspberry, Latham and Glenmoy. As you can see, Latham is a resistant cultivar which has vigorous root system and large number of lateral roots, whereas Glenmoy is the susceptible cultivar with a very weak and unbranched root system. So if you look at the root sections, so Latham has a thicker cortex in compared to Moy. So the first question we asked is Phytophthora penetrating the roots of Latham, or is there some kind of chemical barrier that is preventing the Phytophthora to enter the roots of Latham? So to answer this question, we first treated Latham roots with fluorescent tagged Phytophthora and observed their development through the root. So within 2 hours of infection, we observed infection peg forming, and by 3 DPIs they were developing intercellularly, and by 9 DPI we observed oospores, germinating oospores, which confirmed that Phytophthora is able to penetrate the roots of the Latham. So the mechanism of resistance in Latham may be through induced systemic resistance. So a molecular marker has been identified in Latham, which has been associated with its root vigor and root rot resistance. And this marker has been used actively in breeding by the James Hutton Institute to develop a variety called the Glenmore, which has been released to the market in 2020. And this Glenmore is resistant to Phytophthora root rot. So to have a better understanding of the mechanism of resistance, we focused on the genes that are located up and down from the marker, because as this marker is transferred to the resistant progeny, these genes are also transferred. So we believe that these genes also play an important role in the mechanism of host resistance. So we looked at the genes that were located near the marker and also significantly upregulated in infected Latham. So among these genes, one of the genes that we chose to look further into is auxin-binding protein 19. And as you know, auxin plays a significant role in root growth and morphology. we decided to look further into this gene. So when we looked at the genome database of raspberry, we found multiple copies of this gene, which was located in a cluster, like closely together near the root rot marker. And later on, we found another copy of this gene, which was far away from this cluster. And by matching the sequence of this gene and by doing qRT-PCR, we confirmed it is this gene that was significantly upregulated in infected Latham. So by doing a sequence alignment of the protein of this gene, we found that this gene belongs to a family of protein called germin family, which, so the members of the germin family of protein are involved in the production of hydrogen peroxide, which also plays an important role in plant development and defense. Additionally, many members of this gene family have shown to reinforce disease resistance in host plant. Not only that, genes from this family have been used to develop transgenic plants that are resistant to disease. So one such example is the American chestnut, which has been modified with a germin gene from wheat and the transgenic plant is resistant to chestnut blight. We also looked at the promoter region of this ABP19 gene, and we found multiple auxin binding regions of this gene. So this led us to investigate the effect of auxin and auxin-related chemicals on this gene and on overall root pathogen interaction. So first of all, we started by screening 10 different isolates of Phytophthora with different auxins and auxin-related chemicals. So from all the chemicals that we screened, we found that a form of auxin, that is IBA, and an auxin transport inhibitor, TIBA, both significantly reduced the growth of Phytophthora on plates. So in these graphs, the average growth of all the isolates is represented in the bar graph, and each dot represents a single isolate. We also observed that treating Phytophthora with these chemicals, the sporangia was deformed, and mycelia of the Phytophthora was fragmented and result in darkening and fragmentation. So the suppression of Phytophthora growth by these chemicals may be explained by their chemical structure. So IBA, which is a form of auxin, is commonly used as a rooting hormone, and it has this endoleic heart, which is common to another chemical called camelexin. Camelexin is a chemical that is produced in Arabidopsis as toxins when attacked by pathogen. On the other hand, TIBA, which also is used as a growth regulator, is a benzoic acid derivative which has 3 iodine attached to its benzene ring. So benzoic acid is one of the active ingredients in most fungicides. We have also seen that TIBA, we found it in the literature that TIBA targets a protein called vilin, which is involved in actin dynamics of cells. So we found a vilin-like protein in Phytophthora, and we believe that TIBA may be targeting this vilin protein in Phytophthora and resulting in its restricted growth. So to see whether vilin does have any effect on Phytophthora, we then treated actin-labeled Phytophthora, which means that the actin in the Phytophthora is labeled with a fluorescent protein. We treated them with TIBA and IBA. And after 7 days, we were able to see actin plaques, which is indicated by white arrows here, even in the TIBA-treated mycelium, whereas it wasn't present in the IBA. So we think that TIBA does have have some effect on the actin dynamics of Phytophthora. Next, we wanted to see what effect TIBA would have on root Phytophthora interaction. So we designed experiments where we used hydroponically grown raspberries and treated the roots with different concentrations of TIBA and infected the roots with Phytophthora at the same time. After 7 days, we measured the roots and we found that the mean root volume and mean surface area of these treated plants showed an increased only in the infected samples. We also found that the percentage of dead roots were significantly lower where we had treated the roots with TIBA. So we used root samples from this experiment and we did RNA sequence analysis. And our RNA sequence analysis showed that around 4,000 genes were differentially expressed. And if you look at the heat map, you'll be able to see that the infected and TIBA genes have a very strong expression pattern. We then focused on the upregulated genes in this sample, and we found that a large number of genes were involved in octin-activated signaling pathway. We also looked at our ABP19 gene, and this was also highly upregulated in infected and TIBA samples. So here I have summarized what may be happening to the host roots, how resistance is induced in host roots by treatment with TIBA. So first of all, I showed that treating Phytophthora with TIBA led to fragmentation and degradation of the cell, which will release polymers into the rooting media, which which may directly act as a nutrient for the root growth and also act as PAM or pathogen-associated molecular pattern, which will help to increase its defense response. Secondly, the upregulated genes in auxin signaling is directly involved with root vigor, which will result in improved root growth. And finally, the upregulation of ABP19 genes is resulting in increased ROS production, which is also inducing the defense response in host plant. So how can we use the finding of this research to control Phytophthora root rot? So first of all, so I found out that we can use rooting hormone to control the growth of Phytophthora, and because it is commercially used, it's like high concentration can be used safely. Secondly, we can develop plant activator using Phytophthora cell extract, which may be applied to boost the immune response in the host plant. And finally, the specific targeting of villen of Phytophthora may help us to restrict the growth and development of this pathogen. So with this, I would like to end my presentation, and I would like to thank my supervisors. I would like to thank the University of Warwick for giving me the opportunity to do my PhD. I would like to thank AHTB for funding and James Hutton Institute for everything. And thank you for your attention. And if you have any questions, I'm happy to take them.
Thanks, Raisa. That was really great. I mean, it's one thing to study roots on a model species or a annual crop, but to do it on a woody perennial, that's really quite impressive. So well done. There are no questions in the chat just at the moment, but please feel free to add your questions, because otherwise I'll just have to ask all the questions, won't I? Which you don't want. But basically, first question I have is you mentioned these three potential mechanisms that is causing the tolerance to the Phytophthora, and one of them is auxin promoting root vigor. Do you think this is because that promotion of root vigor, i.e. the promotion of root growth, means that the root is growing away from the hotspot of infection in the soil, for example?
So first of all, I might make it clear that our experiment was not done in soil. So this was only done in solution. So we used a flask, and then it was grown in water with water. The water was supplied with the pathogen. So the treating with TIBA did not lead to increased root length. So it was only the surface area and only the root volume had increased. So I believe that there is some kind of auxin signaling going on, because our transcriptomic data shows that. And also, I think it could be the pathogen that's being degraded. It's actually helping the roots to like boost, because it's not able, by killing it, we are destroying its infection ability, and at the same time those are actually using as a, what do you call, it's also there like sending signals to the root, so that there is something, there's like some pathogen available there, so the root is trying to escape the pathogen as well. So this is also resulting into the root growth. I hope I can answer that question.
I just wonder whether there's some corollary between what you're looking at and what was in the first talk of the day, Bipin's talk, where you've got the ethylene accumulation around the roots and the impact on penetration into harder soils, whether you've got a similar sort of feedback mechanism going on here as well. It might be interesting to think about.
We are so we do plan to do a hormone profiling on these roots as well. So yeah, I think that would be interesting to find out like what hormones are applicable. Yep. Great.
So has anyone got any questions in the audience they'd like to just unmute and ask, or put their hands up if you're tired of typing or whatever? If we've got no questions immediately, then I'd just really like to thank Raisa for a great talk. I thought it was really clear and very accomplished. And again, working on woody perennials, you're very brave. So well done.
Thank you.
Okay. So thanks, Raisa. So we'll move on to the next speaker, who is Professor Gabriel Castrillo from the University of Nottingham. So Gabriel is going to be giving us an insight into some of his recent research on exploring how root microbiome interactions impact stress resilience. So, Gabriel, whenever you're ready, you can take the floor.
Wait a second, I think that I have a technical issue. Do you hear me?
I hear you fine, yeah.
Oh,
and
okay.
we see your screen, but you've skipped on to the second slide, I think.
Yeah, okay, wait a second. Okay, so thank you for the invitation, team, and also Malcolm for giving me this opportunity to participate in this workshop. So essentially today I'm going to talk a little bit about one of the projects we have in our lab related with how the microbiota influence the lateral root development. So I don't want to recreate the wheel. I think that the majority of you know that the root is heavily colonized by microbes. They are everywhere, not only in the rhizoplane, colonizing the meristematic zone, elongation zone, and maduration zone, but also they are in the inside of the plant. So they are inhabiting and colonizing every single tissue of the root. From the meristematic zone, here you have a couple of examples how the microbe heavily colonized the epidermis are also the endodermis. So with high probability, microbes are interfering or influence other processes that are taking place in the root, like lateral root development. What you have here is a scheme of how the lateral root development happens, and it essentially starts for the founded cells in the pericycle that after several divisions form this dome chain structure that need to go through all the layer in the root to form the lateral root. So in this trajectory, with high probability, you're gonna meet the microbes that are inhabiting the root. So in order to study how this influence of the microbiome on lateral root happened, we decided to start doing like a screening of a collection of microbes we have in our lab. Before going into the screening, I want to emphasize that the lateral root development in Arabidopsis is heavily controlled by auxin. So auxin controls the expression of several genes and transcriptional factor. For example, LBD16 on joint NOM, and more recently, other genes related with the suberin deposition in the Caspanian streak that are important for lateral root development. But essentially, is till now, is a question of auxin. So to analyze this, as I told you, we decided to screen a collection of microbes we have in our lab, around 400, that was kindly donated by Professor Paul Essel and Jeff Tungel. Thank you to both of them. So, and this collection of microbes essentially contained the majority of the phylogenetic groups that colonize the root on the natural condition. So to screen and to analyze the influence of these individual isolates on lateral root and essentially root architecture, we designed a simple experiment. So we screened in monoassociation every single element of this collection. So we germinate our plant for 7 days, and then we expose for another 7 days to the individual isolates. And then we took picture and analyze essentially the effect of these individual isolates on the lateral root, on the root architecture. Here you have 5 examples of how dramatic is this effect. So here on the left, you have the no bacteria control, and you see that isolate at L17 or RF27 decrease dramatically the lateral root formation or increase dramatically the formation of lateral root. So this effect is quite obvious and quite evident. So to understand this a little bit more, we quantify at least 24 parameters related with root architecture. So, and essentially what we found is like 80% of the collection had the capacity to modify at least one parameter of this one. But also we analyzed the pairwise correlation, and we identified cluster of highly correlated parameters. So essentially our feature contained redundancy. In order to avoid this, what we did was to select for further analysis the feature that responds more heavily or more clear to the presence of the different bacteria isolates. So, and we reduced from 24 down to, I think there are 14 or 16 different parameters that are not redundant. And using this information, we essentially clustered this, and we identified 24 different clusters that essentially mean that there are at least 24 different ways to produce root architecture where the microbiome is present. I'm going to give you a couple of examples here. For example, you see that the majority of the parameter related with the main root and primary root and secondary root kind of go in the same direction, so going up together and here going down. So one of the first questions that we decided to ask was if these two effects that you see in the primary root and also in lateral root are essentially linked. So when you correlate the lateral root effect, the bacterial effect on lateral root with the bacterial effect on primary root, the correlation was quite weak. So suggesting that these two are not linked. So the bacteria can modify the primary root independently of the lateral root. When you also graph the effect on lateral root and primary root, you see that essentially they don't fully overlap. And they are different, so significantly different, suggesting that actually these two can be independent. Since auxin colonized brutally the lateral root development in Arabidopsis, we asked if this capacity of induced modification in root architecture is somehow linked to the capacity of the bacteria to produce or degrade auxin. But when we went through individual fissure, we realized that actually bacteria that have the capacity to produce or degrade auxin essentially overlap, so there are no differences. Both can modify independently of the auxin, the degradation of synthesis capacity, their root architecture. When we did the same analysis through the individual cluster, and we see no separation, suggesting again that the capacity of the microbiome to induce.
Sorry, there's something on the screen which is covering up the data. So
I'm sorry.
that's okay.
Okay.
Thank you. Sorry about that.
Wait. And we did the same analysis in but individual cluster, and essentially what we saw was no separation, suggesting again that the capacity of the bacteria to produce or degrade housing is not related with the modification in the root architecture. There's something going on here. So in order to study this theoretically housing independent capacity, we select Selaginella as a model for an housing independent branching. So in Selaginella, the branching happened in a different way as Arabidopsis, happened through bifurcation, and apparently this bifurcation is not housing responsive. So we were quite interested in analyze if the microbiome had the capacity to engage with an housing independent branching. So we did that, and essentially we found that there are a population of microbes that induce branching in Selaginella, but also there are a population of microbes that reduce the branching of Selaginella. And here you have an example of 4 bacteria that significantly and clearly induce the branching in this basal plant. So you think that actually the microbiome had the capacity to engage with a branching system that don't, when the housing is not so dominant as in Arabidopsis. And again, we see no separation between bacteria that are able to produce housing or degrade housing and bacteria that have no capacity to influence the housing metabolism in the plant. To go further in this analysis, we identify 4 or 5 classical lateral root development mutants, that some of them are not able to produce lateral root at all. And we quantify the effect of the selection of 100 different bacteria of root architecture. Essentially, what we found is like for the majority of them, we are able to complement the lack of lateral root production. I'm going to show you an example here of the most extreme phenotype we found, that is MF27. And you can see with this bacteria, we are able to complement it R7 and 19, and also ion 184 that on the no bacteria condition are essentially lateral root less. So, and in all of the cases, we are able to increase the lateral root formation, suggesting that somehow the bacteria can complement it, the lack of the capacity of this mutant to produce a lateral root. Here you have more example, and I want to emphasize R7 and 19 after 14 days is able to produce a lot of obvious amount of lateral root in the presence of L344. Another question that we try to ask is this modification in the lateral root formation has consequences at the level of mineral nutrient accumulation. So we determine the ionon, the accumulation of at least 20 different mineral nutrients in the shoot. And actually we determine that for the majority of the cases, we have a significant difference in the accumulation of mineral nutrient, suggesting that this modification have an effect on the nutrition of the plant. And interestingly, some of the elements analyzed positively correlates with the lateral root formation. Obviously, to try to define the mechanism, we quantify the number of primordia at the different stages in the presence of the bacteria in wild-type plants, but also in some of the lateral root mutants. And in all cases, what we see was that essentially the presence of the bacteria increased the total number of primordia, suggesting that somehow the bacteria are rescuing or promoting the capacity of this primordial to form lateral root. Here you have again an example of, I don't know if you see this clearly, but there are more branch enzymes when you have the bacteria present. We determined the correlation between the number of primordia and the lateral root, and this correlation was quite obvious for a primordial at stage 1, 2, and 3 before the differentiation of the apical meristem of the lateral root, suggesting that this promotion can be also housing independent. So in order to be more sure about the exact moment when the microbiome rescue the lateral root formation, we also quantify using DR5 luciferase the pre-branch sites. And you see that in general, the bacteria collection we analyzed don't induce significant differences in the number of pre-branching sites, suggesting that rather than produce more pre-branching sites, what it's doing is rescue the primordia and promoting the transition from be inactive to be active and produce a lateral root. To get more evidence about how this process can happen somehow independently of housing, we also quantified variations in the housing level in the root using this housing sensor, D2 Venus. D2 Venus is taking the D2 domain from IAA 28 and fused to Venus FP. And then when you have the housing present in the system, this protein is sending for degradation. So housing in this case means no fluorescence, and minus housing means that the protein is stable and you have a signal. So using the system, we essentially found that the level of housing in the root doesn't change. And here you have again an example of the most extreme phenotype we found, that is an F27, and the difference in terms of housing accumulation is not significantly different from the no bacteria control. And here you have an example of a positive control when you have IAA in the media, how the root looks like. To get deeper in this mechanism that driven by the microbiota that can be housing independent, we treat the root with two inhibitor, MPA, that is housing polar transport inhibitor, and L-kind, that is housing biosynthesis inhibitor. And essentially what we see was like the MPA treatment abolished the lateral root effect, suggesting that the housing polar, as we expected, is necessary for lateral root development in Arabidopsis. But by the contrary, when you treat the plant with L-kind, the lateral root effect driven by the microbiome remain. In order to define this is no housing and this mechanism can be housing independent, what is the mechanism involved in this effect? So we designed another RNA-seq experiment when we expose again wild-type plants and also mutant to bacteria for 7 days, and then we collect root sample to analyze the transcriptome profile of this sample. We decided to work out of the peak of housing that in our case happened in day 4, so we collect the sample of day 7 with the idea in mind to sort of minimize the prevalence of housing and be able to detect any signal that can be housing independent. For the analysis of the differential expressed genes, we use sort of original system. We use 3 contrasts. In the first construct, we select the genes that are specifically induced in response to the microbiome. And then we filter this selection of genes by the genes that are induced in response to the bacteria in the lateral root mutants. And finally, we only select the genes that correlate with the lateral root production. So using this contrasting, filtering, we identify essentially 2 clusters of genes, one here that positive correlate with the lateral root formation, and another one that is essentially negatively correlate with the lateral root formation. And when we identify was that here in the one that positive correlate with the housing, there are a very nice induction of ethylene-related genes. And when we focus on the ethylene-related gene, we also see that there are a lot of transcriptional factors related with this hormonal response that are getting induced upon the treatment with the bacteria. When we did the same analysis in the different lateral root mutant used, we sort of confirm the same trajectory for these genes. And this effect was more evident when we focus on the genes that are more induced in this treatment. You see that they are significantly overrepresented, enriched for ethylene, but not for housing and cytokinin and FLAG22, other treatments that are able to modify the lateral root. So with this in mind, the next step was to demonstrate that the ethylene pathway needs to be intact to respond to lateral root when the microbiome is present. So we demonstrate this using 3 different ethylene mutants, the double mutant AIM3A1, AIM2, and ATR1. All of them have less lateral root formation when the microbiome is present. And using an ethylene inhibitor AVG, we are able to reduce the lateral root formation in all cases. And in the case of the double mutant N3-AL1, this effect is more obvious. We also were able to sort of recapitulate the bacterial effect through ACC treatment. That is one of the ethylene precursor. And we reduce this effect when we add AVG in all cases. Here you have an example. So this is the Columbia. When you treat Columbia with ACC, you increase the lateral root. With the bacteria, this is more obvious, but both insanely like an explosion of lateral root. And this effect is more obvious in R7 and 19 mutant that is not able to produce lateral root. So when you have the combination of both the bacteria and ACC, you have the density of lateral root dramatically increased in this mutant. Finally, we were able to replicate the results when we are using a more complex microbiome, like a synthetic community of 24 members of a natural community extracted for a natural soil. We were able also to increase the lateral root formation, not only in wild type, but also in the mutant NYOM 184. We also noticed that this effect is independent of the flyelin receptor FLS2. In this FLS2 background, we are able also to see the bacterial effect. And finally, we reduced dramatically this effect when we used the double mutant N3-AL1. We sort of observed the same behavior when we use in stress plant. We treat the plant with sodium chloride and also in minus iron. And we are able to detect again that the natural community and also the synthetic community is able to induce the lateral root formation. So as a conclusion, I want to let you know that at some point in the canonical lateral root formation pathway, there are like a new branch that is essentially controlled by the microbiome that is able to produce a lateral root in an housing independent way, and is essentially controlled by ethylene. I think that this research opened the door to new, I will say, new application of microbial-based strategy. I want to finish with this. Kind of encourage you to explore this a little bit more. So having the key to this mechanism, how the microbiome induced a lateral root formation or changing in root architecture in general, can be interesting for carbon sequestration, also for root pathogen resistance, or obviously for drought and mineral nutrient homeostasis experiment and research, etc. And before I finish, I want to say thank you to the people that getting involved in this research, especially to Mathieu Gonant and Isaid, that they are the leader of this work, and the people on the lab, and my mentors, Malcolm and David Salt. This project had been funded by the University of Nottingham, the Future Food Beacon, and the Leverhulme Trust. And thank you. I'm happy to take the question.
Thanks, Gabriel. Really interesting talk, as usual. So thank you. We have time for a couple of questions. So we have a question from Tanya Galindo. How do you expect this to change in cereal? So are you going to see the same thing in wheat and barley as you do in your experimentation?
What a second. I don't know how to stop sharing that.
So if you go to the top of the screen, you should be able to, there'll be a drop-down menu which says stop sharing.
Whatever. The question is we are expected to see in monocots.
Yeah.
I think that we try in rice and we see some conserve effect for some of the bacteria, but not for all of them. I guess I want to believe yes, but I didn't try.
Okay. We have a question from Doris Vetterlein. So you investigated the isolates separately. However, roots growing in soil show distinct patterns of microbial communities from the tip extending to older parts of the root. Do you think the isolates show similar interactions with root while they are in isolation compared to being exposed to the complexity of the soil environment?
I want to emphasize that the first part of the talk was with mono association, but in the last one of the last slides was with synthetic community and also a natural community structure for a national soil. Obviously, the interaction is not the same when you are in mono association when you are in the complex microbiome.
Okay, I had one quick question as well, which is, what do you think the benefit to the bacteria is of it promoting lateral rooting in the plant?
I think an obvious one is to keep the plant healthy and with more nutrients. I think when you increase the lateral root, you essentially can increase the nutrient. It can be an advantage for the bacteria. Also, you have more surface to colonize. These two are the most obvious things. You keep also the plants happy in soil, and you increase the probability that the host survive, and you can be happy together, I think.
Okay, perfect. That's a really nice way to end the talk. So thank you, Gabriel. That's going to make a start on the main event of the day, which is the medal lecture from Professor Malcolm Bennett. So I've got a few notes here that I want to read, essentially. But I mean, Malcolm's a rare beast, if I could put it that way, in that he's both a root biologist and a fellow of the Royal Society, which is quite rare, I think. But it puts him in good company because Charles Darwin was also both of those, although Charles Darwin only really dabbled in root biology. He didn't really do it properly. So yeah, in good company. But he's got an impressive set of statistics. H-index well over 100, more than 200 publications, approaching 50,000 citations. So Malcolm's one of the most influential root biologists of his era and is in the top 1% of the most highly cited scientists in biology at the moment. So it's a real pleasure to have him here speaking to us. So he's held a number of prestigious fellowships, including BBSRC Professional Research Fellowship, an ERC Advanced Investigator Fellowship, a Royal Society Wolfson Research Fellowship. And his research is focused on the biology of roots and explores how roots grow, develop, and adapt in the soil environment. His group has characterized many of the regulatory signals and genes and molecular mechanisms that control root growth and the developmental responses. And highlights include identifying the molecular and physiological mechanisms involved in gravitropism, hydrotropism, hydropatterning. So Malcolm is also co-director of the Hounsfield facility at Nottingham, which uses X-ray based micro-CT to investigate how these things are impacted in the real world in soil. So that's really important. So, and we have the pleasure of hearing him speak today. So about some of his current research and a little bit about his journey to get here, I think, as well. So I'm really looking forward to it. So over to Malcolm to give your Dundee Root Medal lecture. And this time it's the real award rather than an assumption made on an alumni mailing list. So thanks, Malcolm. Over to you.
Thank you, Tim. And I'd like to thank you and the organisers for nominating me to deliver the ISSR Dundee Medal talk today. It's a real privilege, particularly looking at the roll call of people who have received it in the past. So I'm humbled and very grateful for that. I'd like to talk today about a specific topic, a new topic, which has just been accepted in Science. So we're really super excited about this work, and we'd like to share it with the audience. So it's coming out on November 18th, so another week's time, and it's about how plants sense water stress, and particularly how plant roots do, using a novel mechanism. And just to put this into context, obviously water is rarely an unlimited resource for plants in their environment. So here we have a beautiful image taken in a mangrove forest, and obviously this is really quite unusual. In reality, plants are exposed to relatively dry conditions, as is seen here in the United Arab Emirates, and you can see how they really rely on the delivery of water from the root systems to the aerial plants. This is obviously a pseudo plant. Clearly, delivering water to the aerial organs is challenging, particularly in the context of climate change. And we've really all experienced this summer. In Europe, we've seen the worst drought for 500 years. And this has obviously had a dramatic effect on plants as well as humans. And the net effect of this, and I was struck by this image on the BBC website this summer, the impact of this has been to cause a great real increase in soil drying. And this is an image taken from the Global Drought Observatory. And as you can see, with the exception of West Scotland, even Dundee, the soils in Dundee have been drier. You may be surprised to hear. But clearly, a lot of Europe has struggled, and this is obviously, the drier soil is obviously going to have a major impact on the ability of roots to acquire water and support their aerial tissues. This is not going to get easier. IPCC predictions calculate that Europe is going to be particularly hard hit, particularly Southwest Europe, as you can see here from simulations, in addition to other parts of the world. I think with the exception of Scotland, but you probably won't be surprised by that outcome. So what I'd like to do after that context is understand how water is distributed in soil. And this is a movie that has really influenced me. It was taken by Craig Sturrock at the Hounsfield facility. And Craig took soil from the Sutton Bonington experimental farm and then scanned it using X-ray CT, and then he used false colouring to highlight the soil particles, the water, and then the air spaces. And as you can see, the blue is the water, and you can see how heterogeneously distributed this is within the actual soil. This is at field capacity, so this is what it looks like when soil, the water is readily available for plant use. And as we fly, we take a root cam view of what a root sees when it pushes through. You can see how the water is distributed very heterogeneously, coating the soil particles. And obviously, as the soil gets drier, it becomes increasingly more difficult for the root to acquire that water. Even when there's still water associated with the particles, there'll be in pores which are increasingly difficult for it to acquire. And obviously plants have come up with lots of solutions to counter these problems, these limitations. And this is a lovely review done by Ikram Blilu and colleagues, and they show how desert plants have adapted to water limitation. And in true Arabic style, I'll start from the right-hand side. And as you can see, the cacti have developed a strategy to mine the topsoil. So they have shallow roots. As you can see, in contrast, the legumes have developed a deeper rooting strategy. And plants such as date palms have developed a hybrid approach where they harvest water from both profiles. So clearly, plants' solutions to acquire more water is to increase their root surface area. And one of the reasons that this works so well is that plant root growth and development is closely associated with soil water availability. And it's interesting that Tim mentioned Darwin. Darwin was one of the discoverers of, or he characterized with von Sachs hydrotropism in the 19th century. And we've also characterized it, a great deal of studies. A very talented postdoc in my team is now at Bristol, Daniela Dietrich. And, you know, this is a really interesting adaptive response where plants' roots grow towards water availability. We've also studied a lot of responses which relate to branching. So, for example, hydropatting. This is an adaptive trait originally described by the American group, Jose Dinany, and we have done some work with them, and I'll describe this at the very end. And we've also characterized another adaptive response called zero branching, which controls the branching when roots grow through air gaps, and this will be the focus of most of the talk today. So clearly soil water is able to pattern the growth and development of plants, and this is how plants are able to optimize their root systems architecture. So this begs the question, how on earth do roots sense the water availability in the soil? So these plants are obviously not water stressed, but the plants are able to sense where water is available or not, or better available. So how are plant roots doing this? And this is really, I'd like to take you through a thought experiment about how this may take place. So are there direct water sensing mechanisms where the plant is able to actually sense the presence of water? And there's a very interesting paper that was published in Cell a couple of years ago from Suri's lab, and they described a really elegant prion-based protein domain, which was found on a seed germination regulatory protein. And this underwent hydration-dependent phase changes. So this mechanism is a really elegant mechanism where there are massive changes in water availability, as you'd imagine, during germination. But it's harder to imagine such a mechanism operating, for example, in a fully hydrated cell that would exist within a root that's not droughted. So this begs the question, are there indirect water sensing mechanisms? If you're not measuring water directly through a perception mechanism, are there indirect water sensing mechanisms? And the classic example to think about is, well, maybe as water becomes limiting, there are cell volume changes which cause the actual plasma membrane to pull away from the cell wall, and this actually triggers a change in cell wall integrity, which then triggers cell wall receptors, and that triggers a cascade of signaling events. But again, it's hard to imagine there'd be such large volume changes when a root is fully hydrated. So what other alternatives are there? One tantalizing possibility is that roots somehow measure hydraulic fluxes. And so as the water becomes less available externally, maybe there are changes in the hydraulic fluxes which are able to be detected by the plant. It's very hard to imagine that an aquaporin molecule is counting, given that there are millions of molecules that pass through an aquaporin protein. An alternative mechanism that I'd like to talk about today is something which we call the hydrosignaling mechanism. So this predicts that rather than measure water directly, as I've described, maybe plants are measuring water indirectly using proxy molecules called hydrosignals, which move with water through the plant and trigger developmental responses. And part of the reason I'm proposing this is that many of the root adaptive responses I described before, hydrotropism, hydropatterning, and zero branching, these are hormone-regulated responses. So this begs the question, could hormones like ABA and auxin function as hydrosignals, which are co-mobilized with water and trigger developmental adaptive mechanisms? And to explore this idea, what I'd like to do is discuss some recent work that we've done characterizing this new ABA-regulated adaptive response called zero branching. And to present the data I really would be interested in your feedback. So the mechanism is based on a very simple observation that when roots, and this was done by a very talented PhD student, Emily Morris, and Emily set up a very simple experiment where she grew roots through an air gap. And what she observed when she did this, a 2 centimeter air gap, she would see that the branching pattern was repressed. And this is shown here in this CT image. And you can see here we have a barley and a maize plant. And when the roots grow through this air gap, they cease branching. And this is shown very nicely in the CT image, which I'll show here. And you can see here we have the roots when they're in soil, they're branching very happily. Once they grow through the air gap, they stop branching. And then once they re-enter the moist soil, they start to branch again. So clearly, the branching is associated with contact with moist soil. Is this down to the absence of water in the air gap? Well, by simply replacing water for air and growing the roots through this now water-filled gap, you can see that we can fully restore the branching patterns. So root branching appears to be inhibited when the air is present, when the water is no longer available. So this begs the question, how does root sense the absence of water and block branching? And a clue to this came from a very interesting experiment from Xavier Drey's lab. And Xavier, one of his students was doing an experiment and mistakenly switched off their aeroponic system. And when they came back, they noticed next day that the roots had stopped branching. And when they switched the aeroponic system back on, they started to branch again. And I remember Xavier speaking to me about this in the early 2010s. And my suggestion was check for ABA. See if you can phenocopy this by spraying ABA. Xavier did a very sensible experiment, which after he showed that if he replaced water deficit treatment with ABA treatments, you could suppress branching. He actually measured ABA levels endogenously, and he could see that the ABA levels increased in root tissues transiently during this water deficit stress. So this begs the question, is ABA required for zero branching? The increase in its levels in the root tissues is very interesting, but before we could answer this question, we really needed to acquire ABA mutants and to characterize them. And this is something which a very talented EMBO fellow in our team has been doing for the last 2 years, Poonen Mehra. So Poonen started this project on zero branching, and one of the first experiments she did was to characterize these crop mutants, and she started with tomato ABA deficient mutants. And as you can see, when in an entabulus mutant that no longer makes ABA, you can see here that they are no longer able to repress branching. So this would suggest that ABA really is required for the zero branching response. She's also characterized maize plants, maize ABA mutants, and again, she also sees a failure to repress branching in these air gaps. So this is really interesting because it suggests that in eudicots and monocots, ABA appears to be a very important signal to repress branching during this adaptive response. Working in a crop is all fine and well, but it would be very helpful to be able to do this work with Arabidopsis. And that's exactly what we've done. Poonen very cleverly optimized an agar-based assay, which allowed her to now start to use all the resources within Arabidopsis. So we tried for a long time to develop a vertical system, as is shown on the left-hand side. The problem is roots act in a hydrotropic manner, and they prefer, rather than grow across a gap, to stick to the agar. But by a clever use of an extreme angle of an agar plate, she was able to get the roots to go across this air gap and impose a zero branching response. By doing this, she was able to phenocopy the effects of zero branching and able to show that lateral root branching was repressed when roots were able to grow over the air gap. So by doing this, this allowed us then to start to use all the resources available in Arabidopsis. And to replicate what we've done in tomato and maize, she took an ABA mutant. In Arabidopsis, this is the ABA2 mutant. As you can see, this mutant, just like tomato and maize, is able to, it abolishes the zero branching response, and the roots carry on branching when they're growing across the air gap. So ABA appears to be essential for zero branching. And it begs the question, which root tissue is ABA coming from to trigger a zero branching response? And we did a very simple experiment. We rescued the ABA2 mutant by expressing the wild type gene in just the phloem tissue. The reason we chose the phloem is this is where ABA2 is expressed normally. So we used an phloem companion cell specific promoter, SOC2, and we used it to drive the ABA2 gene. And as you can see here, we're able to rescue this mutant defect. So phloem expressed ABA2 is sufficient to rescue this ABA defect. So this begs the question, why is phloem-derived ABA important for zero branching? And we need to step back 1 second and think about what stress we're actually imposing on the root as it grows across the air gap. So if you can imagine, a root has to grow across this barrier, and temporarily at least, its external water supply has been removed. So it has to rely on its internal resources, and the obvious source of that is from the phloem. And working closely with mathematical modeler Valentin Couvret in University Catholic Louvain, Valentin's developed a beautiful hydraulic model which captures the hydraulic properties, the subcellular, the anatomical, and the environmental scales, and he's able to simulate hydraulic fluxes. And when he's done this and applied this during the conditions a root will experience during zero branching, he's made some really very interesting predictions. What Valentin's model pointed out was, normally when a root is fully hydrated, it's surrounded by water. You can see that the phloem-derived water, shown in red, it stays very close to the actual phloem poles within the steel. But as the root enters the air gap, the external water supply, which is largely coming inwards when the root is in contact with water, when the root is no longer in contact with water, it's growing over this air gap, there's a complete reversal of hydraulic flux. So the water from the phloem now cascades out from the steel up to the outer tissues. It takes approximately 8 hours, Val calculates from his hydraulic modeling, for it to completely replace the original water sources. Now, just to put this into context, 8 hours is approximately halfway across this half a centimeter gap that we use within our assays. So that's around about, the roots are about 2 millimeters in length. So this is what's happening with the water. So essentially what I'm saying is that when the zero branching response is imposed, a stimulus is imposed, there's a complete reversal of flow. So we anticipate that as ABA is made within the phloem's companion cells, it may well piggyback together with water to the outer tissues. So this is our hypothesis, that ABA co-mobilized with phloem derived water during this zero branching response. We obviously want to test this idea, and technically this has not been possible until very recently. With the help of Xander Jones and Jim Rowe at the Sainsbury Laboratory in Cambridge, they've developed a beautiful new ABA sensor, which is far more sensitive than the original version that Xander developed when he was in the US, and is now able to detect levels of ABA which exist within root tissues. And when we use this sensor, this FRET sensor, we're able to see some incredibly dynamic responses taking place. So just to give you an idea, this is a FRET sensor, so we're seeing a change in the ratio, as is indicated by the white coloration of the nuclei, the receptors targeted to the nuclei. And as you can see, as the root grows over the air gap, you can see there's increasing ABA levels are accumulating in the basal meristem. And as soon as the root reaches the other side, it touches the other side of this air gap, you can see the whole system resets. So this sensor is really given us an amazing snapshot on what's happening in this during this adaptive response. We're seeing this amazing transient induction of ABA as it reaches the outermost tissues, and it's occurring within a very defined zone within the root. So why is the basal meristem important? Why do you think the ABA is accumulating in this particular tissue? Well, just to remind you that the basal meristem is actually the site of origin of lateral roots. So if you look at the root laid on its side at the bottom of this schematic, you can see the different stages of lateral root development. So behind the meristem, there's something called a basal meristem, and it's here just as the roots enters the elongation zone, the pericycle cells are stimulated, pairs of these are stimulated to go on to form new lateral roots. And so we anticipate that what ABA is doing here is actually repressing lateral root initiation and triggering the zero branching response. Just to get an idea of what's happening radially, we have also taken images across the different zones, and these are basically cross sections which have been superimposed upon each other, and you can see essentially nuclei. And as you can see in zone 2, which is essentially the basal meristem, you can see how the nuclei, this is the unloading zone for the phloem. You can see how we're seeing ABA changes taking place close to the phloem and the stele, and this is moving outwards. In contrast, closer to the root apex, you can see that the ABA isn't derived from the phloem. In fact, we anticipate is actually coming down from the phloem unloading zone in zone 2, and then reaching these outermost tissues in zone 1 to trigger different responses. As you can see in zone 3, the differentiation zone, there are no changes taking place during this adaptive response. So what we're seeing here essentially is phloem unloading of ABA during a zero branching response. And as soon as the root reaches the other side of this air gap, you can see the whole system resets, and we see a restoration of low ABA levels. We calculate based on the behavior of the sensitivity of the FRET sensor that this represents roughly around about 300 nanomolar of ABA. So the abacus 2 dynamics suggest ABA is co-mobilized with water during the reversal of hydraulic flux. I mentioned that this changes in ABA occurs around about 2 millimeters. That's exactly the same time we predict that water is reaching the outermost tissues based on Val's hydraulic models. So the water and the ABA appear to be co-mobilized with one another. So the next question we wanted to ask is co-mobilized ABA required to trigger an ABA response? And to do this, we took advantage of all the resources that we've got in Arabidopsis, where the whole ABA system has been characterized by researchers such as Sean Cutler. And so we've characterized a large number of these components, the receptors, the phosphatases, the kinases, which make up the ABA signaling machinery. And Poonen has characterized all of these, and whenever we mutate these ABA signaling components, we trigger a zero branching defect. So clearly ABA is functionally required using its canonical signaling pathway to trigger these responses. One particularly important component is SNRK22. If we knock, it belongs to a family of about 10 kinases. If we knock 9 of those kinases out but keep SNRK22, zero branching remains normal. So SNRK22 appears to be the most important kinase for outputting from the signaling machinery. So using these mutants, we ask the next question, well, which root tissues are responding to this ABA that's coming from the phloem? And so we use tissue-specific promoters to rescue SNRK22 expression in different tissues. And you can see here we have one in the epidermis. You can see it rescues in the cortex. It rescues in the endodermis. It rescues. So we were very, very surprised by this result, because essentially complementation by SNRK22 can happen in any outer root tissue. This really confused us. These lines were originally developed by Daniela Dietrich to study hydrotropism. And in that case, we only saw the cortex was rescuing that particular ABA adaptive response. In this case, we see that every tissue, the SNRK kinase can rescue this response. The brainwave came when we realized that maybe there's a common target for zero branching, which the ABA response pathway is targeting. And what that particular target may well be the plasmodesmata. Now, ABA is known to regulate plasmodesmata. Beautiful work by Rishi Balalareru. Her earlier work in a paper in Science has described this in trees. And so we set about asking, is this also the case in roots? So just to remind you why plasmodesmata are important, there are several transport pathways. If I need to remind this audience, there's symplastic and there's apoplastic transport pathways. So in the apoplastic transport pathway, obviously the solutes mobilize through the apoplast until it reaches the Casparian strip. If the solutes enter in the epidermis, they can pass between cells layers through the plasmodesmata. And a great tracer for symplastic transport is something called carboxyfluorosine diacetate. Now, this particular fluorescent dye, once its diacetate's removed, it becomes exclusively symplastic. And so Poonant developed a very nice assay where she dipped roots exposed to different treatments in CFDA to look at their uptake. And as you can see, roots grown on agar plates, fully hydrated, are able to take up CFDA very happily into the inner tissues. But when we expose it to a zero branching stimulus, you can see the actual uptake is severely curtailed, consistent with the idea that the plasmodesmata are closing and blocking symplastic transport. So the question is the ABA machinery acting to gate the plasmodesmata? And this is the evidence to suggest that's the case. So again, we've repeated these CFDA assays where, in the controls, this tracer is very happy to be taken up by roots. And in wild type, when you expose them to a zero branching response, you can see that their symplastic transport is reduced. But in the SNRK mutant, this is the ABA mutant, there's no longer sensitive to ABA. You can see that it isn't able to impose this change, this switch, this blocking of symplastic transport. So this really does suggest that ABA gates plasmodesmata aperture. Now we know that the plasmodesmata closure is regulated by a whole set of different proteins. One, for example, calosynthetases, and there's also proteins called PDLPs. We've mutated members of all these different families, calosynthetases. There are roughly a dozen. When we've mutated these, 9 out of 12 show phenotypes, zero branching phenotypes. Similarly, if we mutate combinations of PDLPs, we see phenotypes as well. So this is really consistent with the idea that if you disrupt plasmodesmata closure by these components, you can disrupt zero branching. So clearly, plasmodesmata closure is important for this response. Just to show you what happens, we've tagged these proteins and expressed them as GFP fusions, and you can see when a root is exposed to an air gap, there is a change in the pattern, and you can see decoration in the blow-ups. In the middle, you can see decorations of the radial cell walls, and we're labeling plasmodesmata. These proteins are ABA inducible. As you can see here, as little as 50 nanomolar, ABA is able to trigger large changes in their expression pattern. And a very important experiment that Poonen did is where we've taken an ABA insensitive mutant, which has a very severe zero branching defect. And you can see here that ectopically expressing PDLPs, so they're no longer reliant on ABA for their regulation, can partially rescue it. So this suggests that ABA controls zero branching by regulating PDLP expression. So our current model is that the transient loss of an external water supply stimulates ABA movement from the flown companion cells. These move out through the plasmodesmata, and they trigger the closure through induction of proteins like PDLP 2 and 3. This then triggers a reduction in lateral root branching. So the elephant in the room really is, well, how is ABA doing this? How is ABA, by gating the plasmodesmata, actually altering branching? And we hypothesize that what they're doing is blocking the inward movement of a lateral root branching signal. And the obvious signal that does this is auxin. Now, to remind you about some work, very nice work done by Leah Band, she's reported a couple of years ago how auxin, rather than rely on transporter proteins, actually relies on plasmodesmata to enter the tissues in the elongation zone to stimulate lateral root development. So for years, I'm sure all of us have been tired of hearing about auxin transport and about how proteins like PINs and AUX-laxes and PDLPs are mediating everything. Well, in effect, what Leah showed in the development paper and reviewed in this new phytologist article is that actually in the elongation zone, in this basal meristem region in which lateral roots are formed, the actual auxin is moving, rather than through dedicated transporters, are actually moving through plasmodesmata. It's moving symplastically. And she showed this very nicely with the auxin redistributions and validated with auxin sensors that this is indeed the case. So our current model is, does ABA act to gate the plasmodesmata and maybe block auxin's inward flux, which is required to trigger the pericycle cells, which give rise to the new lateral roots? To test this idea, we looked at the DR5 reporter in our agar assay, and we looked at it as the roots grow over this air gap to see what happens to the auxin response during this period. And as you can see here, there is a massive change in DR5 expression as we do. And just to make it easier for you to see, if we zoom into the elongation zone, you can see as the root moves across the air gap, there is a build-up, an increase in auxin response in the epidermal cells of the elongation zone. This is very transient, and this is consistent with the idea that ABA, essentially, auxin is not able to enter the inner tissues because of plasmodesmata gating. So our current model is that zero branching triggers a transient auxin response in the epidermis and through gating of plasmodesmata. And just to walk you through the evidence to support this. So what I've presented so far is purely reporter-based data rather than functional data. So if you can imagine with roots which are growing with external water, the auxin which is brought up from the root tip to the elongation zone by transporters like AUX1 and PIN2 it's able to accumulate in the epidermis and then move across to the steel and the pericycle through the action of symplastic transport. So the plasmodesmata are open, and so they're able to enter. When we actually expose the root to a zero branching response, we see a whole cascade of events. The first thing we see is a reversal of water flux, and this causes ABA to move outwards from the steel. This triggers the closure of the plasmodesmata, and that causes an accumulation of auxin in the outermost tissue. So what we've talked about so far is essentially reporter data. What functional data have we got to prove this is indeed the case? Well, for me, the killer, we could be, for example, blocking the inward movement of any signal, not just auxin. So what evidence have we got? It's auxin accumulation in the epidermis that's really the basis for this zero branching response. Well, is blocking radial auxin movement important for zero branching? Well, Poonen did a really important experiment where she took a plant and she overexpressed AUX1. So rather than expressing AUX1 just in the epidermis, where the auxin accumulates, she expressed it instead in every tissue. And when she did this, she was able to synthetically recreate an apoplastic pathway and bypass the symplastic block. And by doing this, she was able to disrupt zero branching. So this is, for me, what I call a killer experiment, because it shows that restoring auxin movement to the inner tissues was critical for a zero branching response. So essentially blocking the movement of auxin to the inner tissues is critical. If we disrupt that movement, if we disrupt the ability to block auxin movement into the inner tissues, we disrupt the zero branching response. And this really elegantly shows that it's auxin that's really important, because AUX1, the carrier we've described in this experiment, is auxin specific. So just to conclude, zero branching is a conserved water adaptive response in eudicots and monocot species. We've shown that using various mutants, ABA mutants. Our hydrodynamic models reveal that phloem acts as a source of water for growing root tissues when the external water supply is stopped, and this causes a reversal of water flux. This causes ABA to flow out, which the ABA is co-mobilized with the water, and this reaches the outermost tissues, and this triggers closure of the plasmodesmata. And this blocks the inward movement of auxin, which is required for lateral root initiation. Strikingly, I think the work shows that once root tips regain contact with moisture, the ABA response is rapidly attenuated. And this shows you how dynamic these responses are. And we were completely ignorant of them until we had these wonderful tools that Xander has developed and Jim. And I hope this study has revealed that how roots adapt their branching patterns to heterogeneous soil water conditions. And what they've done, which is very clever, is link changes in direction of hydraulic fluxes with this dynamic redistribution of ABA and auxin signals. So to answer the question I posed at the very start, do roots sense water availability via an indirect mechanism? I hope I've convinced you that indeed this is the case. Plants appear to be using ABA as a proxy for outwards water flux, because which involves the co-movement of water and ABA. And we term this signaling mechanism hydrosignaling. So ABA and auxin are acting as hydrosignals. So it appears in this particular context, plants are not measuring water directly, they're actually using these hydrosignals, which in this case are hormones, but could theoretically be other types of signals. I'd like to thank a lot of people for this. I've acknowledged them during my talk, but I'd like to particularly highlight the role of Poonen, who is a remarkably talented young researcher. I hold out huge hopes for Poonen, and She fully deserves the co-corresponding authorship on this paper. She really was an intellectual and practical driving force behind it. I'd like to just finish off very briefly, Tim, just to talk about the journey that we've taken. What really got me here, I wondered you might be interested to look at, what's pointed me in this direction of research. I've had a lifelong fascination with plant roots. Who can I blame for that? I have to blame my PhD supervisor, Julie Cullimore, who is sadly retiring early next year. She was an amazing supervisor. I was the first PhD student, and it was a privilege to be supervised by her. I did a PhD on root nodules, and at that point I was completely suckered. Roots, the hidden half of plants, completely captivated me. My interest in water and root development really was quite late. We did work with Christophe Morel's team and Tony Schaeffner, and characterizing the role of aquaporins during lateral root development. And in our team, I worked with a very talented researcher, Benjamin Perret, who is another EMBO fellow, and also Leah Band, who's still at Nottingham. I'm very proud of Leah. She's now associate professor. Benjamin is now a senior researcher, group leader back in Montpellier in France, and was an ERC recipient. So my interest in water and hydraulics really was teased by this. For years, I was interested in cell walls and chemical signals, and the idea of hydraulics was really quite exotic and challenging. But this really whet my appetite. We carried on and started to look at how roots grew in soil. And this really was a revelation for me. Working with Sasha and Craig in the Hounsfield facility, it's really opened my eyes to the importance of soil and understanding the way that plants interact with the soil matrix and water. And Jose Dinany was a huge influence on me. Jose described to me how roots are responding to environmental signals. Up to this point, all I did was very much characterize the development of lateral roots and looking at their developmental responses, and not really considering the importance of the environmental signals which are controlling these pathways. So Jose, Sasha, and Craig really played an influential role of me to really think about the environment and how that impacted plant development. Later on, we figured out the mechanistic basis for this, and this was working with really talented people at Durham, Ari and Beatrix. Beatrix is now in Edinburgh, and members of our team, Jason, Daniel and Nikki, and we were able to elucidate the molecular mechanism which was driving this. We also started working during this period with Xavier Drey on a completely different mechanism. In this case, this is zero branching, which I've spoken about, and Emily Morris was the PhD student doing this work. And as I've described to you just now, we've characterized the mechanism behind zero branching, and this is very much, I'd like to flag up two people in particular. Valentin, whose hydraulic modeling really helped us understand the hydraulic fluxes which were driving this process, and Poonen, who has been a complete driving force behind this. She generated all the data I presented and much more in a 2-year period, which is really quite extraordinary. I'd like to just quickly mention that we're very, very interested in looking at the additional scales to go beyond root systems architecture, to start to look at root anatomy. And as many of you know, anatomy has a major effect on root-water relations. So understanding the genetic control of anatomical traits like xylem vessel size would be extremely important for developing future-proofing crops to climate resilience. And this work has obviously been pioneered by Jonathan Lynch, who was the first recipient of the ISSR Dundee Medal. And Jonathan has been a great supporter to develop laser ablation tomography facilities at Nottingham, which have really helped us perform anatomical phenotyping. And using these tools, and this is an example of one done by Dylan and Brian Atkinson, we can actually start to really uncover anatomical features which are really can form part of a breeding program to start to breed for anatomical traits. And we're using this in projects sponsored by the Royal Society and the French funding agencies with colleagues in France and Senegal. And this year, I had the privilege to work in the field. That's me in the hat, digging up the first plant from our field trial. And that's Darren Wells, who leads the laser ablation tomography work at Nottingham. And we're working with colleagues, Alex and Laurent from France and Gido and our in Senegal. And we're using these cutting edge phenomics approaches to really start to understand the role of anatomical traits. And I think this is a really exciting area, which hopefully will make some really big steps in the future. And finally, I'd just like to have a big thanks to all the root group members at Nottingham. We've got 10 groups, research fellows, from small teams to big teams. You've heard from some of them today. It's a fantastic environment, and I'm very, very grateful for their support and enthusiasm, particularly over the last few years with COVID. I don't know what I'd have done. I would never got through without their support. So thank you so much to the team. This award is very much to the team rather than to the individual. Thank you.
Thanks, Malcolm. That's really great. Please, everyone, express your appreciation for Malcolm's talk. It was really fascinating. And of course, it's generated lots of questions. So there's lots of opportunity for us to ask some questions now. So what I will do is start with Dhin Islam, who's asked the question, zero branching. How can you confirm roots stop branching due to a lack of moisture? And if I add to that a little bit, how does this interact with what Gabriel showed us about the impact of bacteria on lateral root production?
Sure. I mean, I think the experiments which we've done in natural soils showing, which is going to have a comprehensive microbiome, and in the air, there are no lateral roots formed when you put them in. When you place them water there, you can completely restore it. I think it addresses that question. That essentially it is entirely water based, so a limitation. Rather than, for example, a defense response, I would argue against. We've done a lot of these experiments, obviously axenically in clean plates, and we see the same kind of responses. So we know they're not dependent on the microbiome. The microbiome could well have an impact, and it's something that we really want to work with Gabriel on, and that's one of the benefits of having this expertise within our team. So we're these are kind of experiments that Gabriel and our team are doing. We're looking at this kind of nexus between the soil, the microbiome, and the plant itself. And so, yeah, these are obviously experiments we're exploring.
Great. So next we have Jambo Shen, another awardee. So it must be quite late with him, I would imagine, in Beijing, but he's asking some questions, which is there a common pathway for root foraging to soil moisture and for nutrients such as nitrogen and phosphorus? So both water and nutrients that can stimulate root branching. So any comments, particularly with respect to how we might produce better crops for these kind of things?
That's an interesting question. I mean, I'm really kind of Catholic in what I would call a hydrosignal. So as long as the molecule moves with water, is able to move, and obviously plasmodesmata are highly non-selective. These molecules, it could be a very broad designation. hydrosignals, and as long as they're then sensed, there's the sensory machinery within the cells, these molecules could well trigger responses. The ability of nutrients to do that it's entirely plausible mechanism. And in terms of how you can improve plants, that's a really interesting question. We know that, as Bipin's shown very nicely, that signals like ABA play a very important role during things like compaction resistance. We anticipate a lot of these responses are going to be linked. So things like hydrotropism, zero branching, compaction, they're all ABA regulated processes. And we suspect there'll be common mechanisms. And it's something that we're starting to discover. We're identifying several mutants which have defects in several of these responses. So we think that there is probably a common water sensing mechanism in roots, which involve ABA. So it's an area which we're very excited about, and we're actively exploring.
So Glyn Bengoff says, great presentation. Does the increase in ethylene diffusion rate away from the root surface in the air gap have any interaction with the ABA auxin? Can you exclude any role of mechanical pressure on the root tip or root branching?
That's a great question. And as far as I know, Poonen has looked at ethylene mutants and doesn't see any differences. So we're pretty confident that, ethylene perception is not really required for this response. We think that this isn't requiring gas diffusion as such, as the compaction story does. But it's not something that we've done a lot of work on. So it's something that, I agree it would be good to do in a follow-up paper. Yeah, great idea. Thank you.
And Adam Price says, beautiful stuff, Malcolm. Such an elegant description of plant biology. But here's a question anyway. Do you think the primary function of this response, the original evolutionary drive, is regulation of branching or perhaps preventing water loss?
That's a really cool question for lots of reasons. Because what we're essentially proposing is the root is, when it goes over the air gap, it's essentially going from a symplastic uptake mechanism to an entirely apoplastic pathway. And that is really interesting. And we're currently doing all the transcript profiling to see, is it, are we seeing, when you close the plasma desmata, you're going to have to upregulate all these different types of transporters to start moving all these other molecules to the cells to carry on growth. And it's something that we've used the traces that would suggest to be the case. Clearly, the plant prefers to have symplastic transport. So as soon as it goes back on the other side, it reverts to symplastic transport. I've puzzled about water loss. That's a really interesting question. I haven't got a proper answer. I've asked lots of experts about it, and they haven't given me an answer. So I think we've got to go away and measure it. So I haven't got an answer for your question. I've asked people like, oh God, what's the guy called? A famous person in Montpellier who does a lot of water stress work. Yeah, Christophe, and I've talked to several people, and I haven't got a proper answer. So I don't know whether it's a water saving mechanism or whether it's essentially altering their architecture. Is the short answer.
Okay,
thank you.
So Vincent Espinoza Hernandez asks, Malcolm, the challenge is there for a new generation to focus the study on root paradigms. Congratulations. So it's really a comment that you've set up a whole lot of challenges for people to pick up.
Thank you.
Yeah, well, there's lots. Sorry, Malcolm, carry on.
No, go on, please.
No, I was just going to say there's lots of congratulatory remarks in the chat, so please have a look at those. I have one last question before we go to the medal, and that is, if you could design a root idiotype for a crop to cope with this extreme variation or temporal or spatial variation in water, what would it include?
Well, if I was Jonathan Lynch, you'd be hearing steep, deep, and cheap.
Mm-hmm.
Yeah? And you'd be talking about steeper roots and things. But I think ecologically, if you read the ecological literature, actually that idiotype works. It's great under some soil conditions, but it's not great under others, and that's why I deliberately put the desert plant example up there, because a lot of desert plants actually have very shallow roots to capture the intermittent rainfall. So I think the date palms are onto something, because essentially they've hedged their bets. They've gone both ways. As I mentioned, I do quite a lot of work in Senegal, and one of Laurent Laplace, I remember some of his staff came and worked in our team, and we were just amazed by pearl millet, the crop they work on. This thing goes straight down. They planted just at the start of the rainy season, and this goes straight down. It doesn't branch. So it depends on your conditions. It's all down to your waterfall patterns and your soil types. So I don't think there's a one size fits all.
And I guess plasticity has to fit in there somewhere as well, right? So, and that's a big challenge.
Absolutely. Understanding plasticity, and I think hopefully that's what people have got from this talk, is that we are starting to tease apart. What I've talked about is not drought. I've talked about one root tip making a decision. Do I branch or don't I? And devolved decision making. And I think this is really, really important. So I think what we've described here essentially is a mechanism for plasticity.
Yeah. So there's plenty of work still to do, right?
Absolutely. I've still got 10 years in me. They'll all grown when they hear that.
Yeah, fair enough. Okay, so maybe if everyone can anyone who wants to can put their video on, and we can congratulate Malcolm in a appropriate way. I believe also that David Salt is on hand.
He's here.
There to present the medal on our behalf. And before you get too excited about the medal,
come round so everyone can see you. Yeah, we filled the room.
Yeah, I can see that. That's brilliant. So yeah, don't get too excited about the medal. That's all I can say. But
Well, my wife says she deserves this medal.
Well, from what you said the other day, how many years?
44 years she's been with me.
Yeah.
Since we were 14. So that's quite. She definitely deserves a medal.
Yeah, well, yeah. We'll get it engraved in her name rather than yours then. But yes, please. So anyway, over to David to actually hand over the medal on our behalf.
Well, here I am. It's really quite an intriguing style of ceremony, but nonetheless, it does not undermine the significance of it. So, I mean, it's great pleasure to give this medal to you, Malcolm. Dundee Medal for sustained effort in root biology, making significant contributions. I think that Malcolm's record speaks for itself, but clearly he's made us a sustained an effort in working in root biology as he has keeping his marriage together. And I think that we all know the kinds of work that Malcolm's done over the years. But I think I think it's quite nicely summarized in the fact that Malcolm's published 6 science papers, the first one in 1996. And I wouldn't say the last one, but the next one just to come out. So 6 papers over that time, 20 odd years. It's a remarkable contribution. 2 Nature papers, 17 PNAS papers. So a remarkable effort. And I think it not only speaks to the quality of Malcolm's work, I think importantly, it speaks to the sustained effort that he's put into this work over the years, not only in making great discoveries, but also nurturing a whole set of great talents, which I think is also really, really important. I think one last thing from me is clearly Malcolm is not reaching the end of the road. I mean, his publication record and his grant capturing record has really just taken off in the last few years. I mean, 3 science papers in 4 years and about 1.7 million quid from BBSRC. So I mean, it got more great things in him even though he's been telling me he's just upgraded his camper van. I'm not expecting him to be going off into the sunset anytime soon. So I'm going to be quiet now, and I think we all need to get down the pub. So anyway, great pleasure to award Malcolm this spectacular medal. You probably can't see it, but it's been made by a medalist in Edinburgh. I didn't even know medalists existed, but apparently they do, and they make some cracking medals.
And if you look on the back, it's engraved in your name as well. So congratulations.
I really appreciate, I'm humbled
David, it's a really great to get this from you, because you're someone who I hold up in great esteem, and it means a lot for you to give it to me. You've had a huge impact at Nottingham since you've come from Aberdeen. Aberdeen's loss is our gain, and it's been such a pleasure working with you, and all the great things you did with the Future Food Beacon, and the generations of people you've developed, and the support you provided. And that's, I guess that's something that we have in common. At our stage in our career, supporting young, talented researchers is the best part of the job, and mentoring them. And today you've heard about Poonan Mehra. It's not the last time you hear her name. She's an extraordinarily talented individual. And it's just been such a pleasure working with her on this particular topic area. You've also heard about Rahul and Bipin and Gabriel. These are, people that have been mentored by David and I and others. And it's just honestly, every day feels like I'm opening a chocolate box, because I work with extraordinary people, and it's just such a privilege. It really is. And so thank you so much. This is very much a team award, because there's no I in a team, to use a horrible phrase, but it's true. And it's very much a team award to the Root Group in Nottingham. So thanks very much, Tim. Thank you for the ISSR board for recommending me, but I'm taking this very much as a team recognition. So thank you.
Great. Well, maybe you can have it once. Everyone can have it for a week over the next year or something, and cherish it in that way. But yeah, okay.
There's no hole in it, so I can't wear it like a medallion. David,
we can get that sorted for you. Yeah, we can get that sorted for you, Malcolm, if you like. But
I got a drill at home. I can do a local job here.
But I need a chest transplant then.
Oh, right, yeah. Well, we can get you something else as well. But yeah, thanks ever so much. And I mean, people, please feel free to stay on and have a quick chat if you want. But otherwise, I think the guys in Nottingham want to get to the pub. Okay. Fantastic. Well, thanks everyone. Thanks for joining. I think it was a really interesting afternoon. So great.
Thank you very much, Tim.
No worries.